I think about both the bridges we build and the community we have. When Sangeeta and I took over organizing this symposium 6 years ago, building community here locally with our colleagues interested in liver disease was really one driving force behind it, because there's so many of us here interested, and we also wanted to make sure that we engage and excite our trainees. And so it's good to see so many of you here, and I don't know how many there are online, but I'm sure there are a lot. Communities also obviously goes further, and I think what's so important for us is that we engage not just with liver disease as an entity, but also with our patients who have that disease. And so as we bridge, I think it's important to highlight that ultimately our basic science needs to reach our patients. And I think especially with our first speakers who will enlighten us about their work on hepatitis C and others, I think it also highlights how our society has failed us in translating those basic science advances into cures at the bedside. Because as of last year, we still have more patients infected with liver disease, with hepatitis C, than we are curing, even though we have the most advanced therapy. And so for us to reach out to our patients, to engage them, to work with society, with our politicians, to actually make that happening is just as important. And that's why I'm really excited that we can partner with the American Liver Foundation, who's not only great in supporting our research with grant funding, but also are fearless and a force as advocates for our patients to be treated well, and so that we do not marginalize patients and leave them to their disease. So I'm very grateful for that partnership too. And before I hand over to Sangeeta, I want to highlight one thing. We made this pact when we started this series, organizing it, that we would have gender balance at every single meeting. And if you look through our list of speakers over the last 6 years, I think we're within one person being balanced between male and female speakers. And I don't think there's another symposium series that has achieved that. And so I'm very excited about that too, that we can be leading and pointing into the future there as well. And now I hand over to Sangeeta and wish us all a great day.
Thanks, Wolfram. Hi, everybody. So there's not much to add, but just a couple of things, just to say that Wolfram and I got involved in 2017. We accepted the baton from Wynn, who received the symposium as somewhat of a present from the American Liver Foundation some 32 years ago for his extraordinary contributions. And he and I had been participants in this meeting as trainees and met, Wolfram met Tom Starzl. I first met Charlie Rice, who you'll hear from today at this meeting. And we really envisioned a future where more and more trainees would come into this field and bump into each other and realize how exciting the basic science translating to clinical liver disease is. So that's our goal for today. And this hybrid format is really an experiment to try and broaden our reach, an experiment that we learned from the pandemic, and we're going to keep learning as we go. As of this morning, we had 500 registrants, 10 speakers from around the world. We have micro talks from trainees, really to highlight their visibility. 16 poster judges from academia, hospitals, and industry. We're going to thank everyone again later, but I wanted to start the day by thanking Wynn, of course, for his inspiration and example, and Heather Fleming, who really worked together with the ALF team to make this happen. With that, I'll just do one little bit of Zoom housekeeping, and then we can get started with the fun science, which is for those attending by Zoom, hello, please use the chat for questions, which will be passed along to our session chairs during Q&A. And for those who are live, we have mics on the side of the room. Please silence your phones. Feel free to tweet about us because we want everyone to know this liver symposium is happening, and enjoy the science of the day. So welcome. With that, I'll have Kay come up and introduce the first speaker.
Good morning, everyone. My name is Kay Alcedo. I'm a postdoc in the Camargo Lab at Boston Children's. Yeah. And I'm here with Constantine to chair the first session. So Constantine will do the Zoom, and I'll be here monitoring the first session. So I'd like to introduce the first speaker today, and that is Dr. Harvey Alter. Dr. Alter is a distinguished NIH scientist emeritus from the Department of Transfusion Medicine at NIH. As one of only 23 NIH scientists to hold this title, Dr. Alter has played a key role in the discovery of two hepatitis viruses, Hep B and Hep C. In the long-term perspective studies, Dr. Alter helped define the natural history of HCV infection and prove that this frequent progression from chronic hepatitis to cirrhosis and liver-related mortality. He has received many distinguished awards, and I can go through all the accolades, but we will be here the entire day. So I just want to wrap up that one of his many accolades is that he has won the Nobel Prize in Physiology and Medicine in the topic that he's going to discuss today, and that is entitled Hepatitis C, Then, Now, and Beyond. Let's welcome Dr. Alter.
Can I advance the slides? Okay. Well, thank you very much. And starting right by, I want to thank my young mentor, Arias, who runs the most amazing bridging science course at NIH, the best course ever. And this is sort of the starting point of that course. It's been wonderful and great to know when. So in light of time, this is my talk today. If you look at the title, this is really what I'm going to address. And unfortunately, I can't advance my slides myself, so there'll be little pauses here. But we always have to now do financial disclosures. We have the next slide, please. As you know, next slide, people, no, this is going to not work. Go back. People that work at NIH only dream about financial disclosures, but I do have some other disclosures that might be of interest. Next slide. Next. So my guiding scientific principle, and I can't see my thing either, is that to steal from one is plagiarism, but to steal from many is research. Secondly, nothing I say reflects the position of U.S. government. Next. Which instantly gives it great credibility and relevance. Next. Long before AI, I realized that my intelligence was artificial. And lastly, my memory is not as sharp as it used to be, and also my memory is not as sharp as it used to be. Now, next slide. This is the most important slide I will show for the young people in the audience. I was given this as a congratulatory card when I got the Nobel Prize, and it said there's no elevator to success. You have to take the stairs. And this is so true. This is a research or medicine, clinical medicine. It's a step-by-step process. And today I'm going to tell you some of the steps. There are 50 steps, but I only have time to tell you 20. But I will go through them. But remember this slide, because you don't jump to the end point. You really have to climb your way up in research and then get there. Next slide. So we started out to address the question of how frequent is post-transfusion hepatitis, and if possible, what are the main causes of post-transfusion hepatitis? And we did this by setting up a prospective study of open heart surgery patients because they received a lot of blood in those days, and because they were not immunodeficient in general, so that they would develop antibodies that we could measure. The first striking point was we found that when you looked at people prospectively and you measured liver enzymes, didn't wait for clinical jaundice, but you measured liver enzymes, and we did this every 1 to 2 weeks for 3 months, and then every month for another 3 months, that the frequency of liver enzyme elevations, which we equated with hepatitis, was around 30%. This was a startling figure. And the main reason for this, we found, a study John Walsh did, was that it was all due to the donor source, that if you received at least 1 unit of paid donor blood, there was a 51% chance of developing hepatitis, whereas if you received only volunteer donor blood, it was only 7%. So we didn't think we could get enough volunteer donor blood to ameliorate the situation, but we eventually did. So in 1970, we went to an all-volunteer donor system, look at that downward arrow, and also introduced the first very simple test for hepatitis B surface antigen. And that caused a precipitous fall in post-transfusion hepatitis from 30% down to just under 10%. And nothing we've ever done since that time has been as potent because we never had rates this high again. So that's where, next slide, please. Next slide. In 1973, Abbott Laboratories developed a more sensitive test for hepatitis B, and we went to our stored samples and was surprised to find that only about 25% of post-transfusion hepatitis was related to hepatitis B, and there was this big non-B entity looming out there. In 1975, next slide, Feinstein and Kapickian and Purcell, who were at NIH, using this immune electron microscope, which is still in the hallway of building 50 at NIH, they discovered the hepatitis A virus. And since Steve Feinstein and Bob Purcell were our collaborators, we immediately sent our samples, our non-B samples, to his lab, Steve's lab, and not a single case of non-B hepatitis was due to hepatitis A. So it was then in a brilliant step of deductive reasoning that we said if these cases were not A and were not B, we'd call it non-A, non-B hepatitis. I apologize for that term, but we were pretty certain we would discover this virus very soon and then rename it hepatitis C. But we didn't know at that time whether it really was a virus, and we didn't know, if so, how many viruses might be involved in non-B hepatitis. So now we had, next slide, now we had non-A, non-B, and then the next step in this process was, next, was the chimpanzee. The chimp was invaluable in our studies. We're not allowed to use chimps anymore, but many felt they should have received the Nobel Prize because we learned so much from transmission to the chimp. We're not allowed to do this anymore because it's thought to be inhumane, but none of the chimps got sick. And indeed, they love being in our studies. You can see they're lining up here to sign their informed consents. But we learned by transmitting, next slide, that we could transmit hepatitis to the chimp. Again, just measuring ALT elevations in the chimp, that we could transmit from patients with either acute or chronic non-A, non-B hepatitis, or from totally asymptomatic blood donors who had been implicated in transmitting hepatitis to their blood recipients. Next slide. The next big thing and the next step on this staircase was this patient, Mr. H. Mr. H, we learned more from this single patient than from any group of patients. pointing out that if you really intensively study one patient, even though you can't publish one patient very readily, but you can learn an enormous amount from one patient who's carefully studied. Well, Mr. H liked to blaze trails on mountains. And one day he was up on blazing a trail and he had a cardiac arrest. And he would have died on that mountain, but his wife was with him and she knew CPR and she resuscitated him. He made his way down to an emergency hospital and eventually made his way to NIH, and he entered into our prospective study. And in fact, I followed him for 30 years after that initial episode in 1977. Shown on the left here is his clinical course, in which, just pay attention to the blue curve, that's his ALT elevations. And you can see he had normal levels until about week 5, when the ALT started to go up, and then it rose precipitously up to a peak of 2,112. At that time, he was jaundiced, he was very sick, but turned around very rapidly and got better. His ALT levels came down just as precipitously, and he improved clinically. At point A, we obtained a plasmapheresis unit from him. We put that into a chimp. We put just 1 ml of that unit into a chimp, and the chimp developed hepatitis, as shown in the upper panel on the right. Later on, much later on, a decade later on, PCR came along. And we were able to go back into these stored samples. And if you look at the yellow line now, that's his PCR, that's his HCV RNA level. You see that at week 0, he's negative, but at week 1, there's a little blip of HCV RNA. That was his donor virus getting into him and being diluted out by his blood volume. And then that seemed to disappear. And then around week 4, HCV RNA began to rise again as the virus was replicating in the liver and then came out into the bloodstream. You see that the PCR went up very rapidly as well, to 3 times 10 to the 7 copies per ml, which is identical to a chimp infectious titer. That's your thing. Oh, thank you. But note too that as the HCV RNA came down, the ALT came down right after it. So, but now we had a titered infectious inoculum. Bob Purcell took that material, put it into other chimps. We knew exactly how many viral copies there were. And we had the chimp model. That allowed you to manipulate that inoculum. And without showing you the data, we did, Steve Feinstone did chloroform extraction, showing that the virus disappeared when you treated with chloroform. Therefore, the virus had a lipid envelope. We presumed it had a core inside. We did filtration studies, again, filtering material, and then putting that into chimps. And we could show the virus was quite small, 30 to 60 nanometers in size, lipid enveloped. So it already narrowed the possibilities down to these small RNA viruses, alpha flaviviruses. Could have been a hepatitis B-like virus, but we had a lot of evidence that it was unrelated to hepatitis B or to Delta. So it's either going to be a new viral class or a small RNA virus. And as it proved to be a flavivirus later on. Now, we kept trying to develop a test for this to see the virus, et cetera, but we couldn't do it. Somehow it was just very, very elusive. So we concentrated on the patients. What happened to people who had non-A, non-B hepatitis? So a lot of people thought this was just a benign transaminitis. But with the liver group at NIH, we biopsied 39 patients and found that most of them had very mild to moderate disease, but 10% already had cirrhosis the first time we biopsied them, and 13% had what was called chronic active hepatitis that would have evolved into cirrhosis. And then we did rebiopsies at varying intervals after that, and found that most patients were pretty stable, or you actually seemed to improve, maybe by sampling error. But 25% progressed to cirrhosis under observation. So in the end, we had 8 out of these 39 patients, or 20%, who developed cirrhosis. And as you see in the bottom, 3 of them died of liver failure. 3 of them had very severe liver disease when they died of their underlying heart disease. So we now knew that this was a virus that could cause progressive fibrosis, and it could be a lethal fibrosis. This got everybody interested in non-A, non-B, and suddenly the field emerged. So we had now the clinical severity. We tried everything. We tried all the known serologic, enzymatic, radioimmune assays. We still couldn't find an antigen or an antibody. And it's hard to find an antigen when you don't have the antibody, and vice versa. But molecular biology hadn't yet emerged. This was still early 80s. But it was kind of frustrating because we were stuck with this non-A, non-B nomenclature here. So at that time, I wrote a poem of frustration called "I Can't See the Forest for the HBAGs." And when I think that I shall never see this virus called non-A, non-B, a virus I cannot deliver, and yet I know it's in the liver. A virus that we often blame, but what exists alone by name. No antigen or DNA, no little test to mark its way. A virus which in our confusion has forced us into mass collusion to make exist just by exclusion. But is it real or an illusion? Oh, great liver in the sky, show us where and tell us why. Let us see this elusive virus. Send us thoughts that will inspire us. If we don't publish soon, they're going to fire us. So, well, that poem, which I read at a national meeting, had a lot of influence because very soon after that, the Chiron Corporation that had been working in secret cloned the hepatitis non-A, non-B and called it hepatitis C. They did this by a technology that today would be quite simple. But in 1985, 86, 87 was a real breakthrough in molecular biology because, first of all, they took known infectious material that had been proven infectious in the chimp. They pelleted down any potential virus, extracted the RNA, extracted it. There we go. Reverse transcribed that into cDNA, cut that up with enzymes. And then the next important step was they used a newly discovered phage, GT11, which would not only express the genome of the virus, but would express any protein that was coded for by that genome. They infected E. coli with the virus. They grew the E. coli in culture. Gliced the E. coli onto filter paper, and then made the third important step was they assumed there would be antibody there, even though nobody had found an antibody. And they screened these clones with the antibody. The story goes that they screened 6 million clones before they found a single one that was positive. They did this over 5 or 6 years. But when they found a clone, they were able to subclone it, then to express a small protein, then to walk the genome and express a bigger protein, and then develop an antibody assay. And when they did that, they called this hepatitis C, and they came to me for a panel. Oh, this is Michael Houghton, who was the leader of this group at Chiron. I guess because I only had 20 minutes, I cut out the panel. But anyway, we showed that hepatitis C and non-A, non-B were identical, that the patients who got hepatitis in our study were negative before transfusion, became positive for antibody after transfusion. We found that we could detect a blood donor that was positive in 90% of these cases. And so we now had an assay that we knew if we applied that to the general blood donor screening, we might prevent 90% of hepatitis. So this is the 30-year study. I showed you the left-hand side before. Over the years, the rates of hepatitis came down because the blood usage shown in the squares had come down from 17 units to about 7 or 8 units per case. So by 1990, just before we introduced the first test for hepatitis C, we were already down to a rate of 4.1% from the initial 30%. The first generation test for antibody to hepatitis C brought it down to 1.1%. And by a second generation test came out in 1992. And by 1997, we had reached 0. And we now followed about 1,000 more patients after this for HBV DNA and for HCV RNA, and I haven't had a single case of hepatitis since 1997. We could estimate from the incidence figures along the way here that blood transfusion may have caused almost 5 million cases, 5 million infections, not cases, 5 million infections in the 2 decades from 1970 to 1990. Not all of these patients would have gotten hepatitis. Many, if they got hepatitis, would have died of other causes before they had bad outcomes. But nonetheless, 5 million people were set up to possibly get cirrhosis in the long term. Conversely, once we introduced this test, we could predict that we probably prevented another 2.4 million infections in the next 2 decades from 1990 to 2010. Now, during this whole time, I was a government employee. And government employees get a bad rap as being lazy and just living off the government dole. So I just want you to know that I always gave 100% of work. It's just that it had this peculiar distribution. And I'm not even sure what day today is, but any case. So the reason there's so much hepatitis C around is that 75 to 85% of people who get hepatitis C develop a persistent infection. And we looked into why that might be true. And this one slide illustrates why. This was done by Patricia Farsi, a collaborator of mine at NIH, where she took Mr. H, that same patient, and did 105 clones of the patient and sequenced those clones. And 57% were identical. That was his dominant strain. But at the same time, there were 19 other variants, slight variants that were very close, but still antigenically distinct. So that the antibody that attacked the primary virus would be overcome by any one of these other variants. And the virus was always a step ahead of the immune response, and the infection persisted. Then we got interested in why do some people get over the infection and other people go on to chronic infection, and why do some of them get very severe disease and some get very mild disease? So we looked at what we called slow progressors versus rapid progressors. The slow progressors shown in yellow, the rapid in blue. And what we found is that rapid progressors had high levels of MCP1, a monocyte chemotactic protein. This is a pro-fibrogenic chemokine that both attracts stellate cells, the cells in the liver that lay down collagen, and induces the stellate cells to make more MCP1, setting up this continuous loop of stimulation of stellate cells and the laying down of collagen. In contrast, the slow progressors had increased levels of interferon gamma and MIP1 beta, and these are the kind of the good cytokines that decrease inflammation. What I didn't show you here is if you look at the yellow line, the slow progressors seem to contain the virus. So there was a rapid rise, and then a diminution, and the virus almost goes away, but then comes back again as the yellow line goes back up. This was the initial virus being contained by the immune response and a second variant coming out and causing the persistent infection. In contrast, the rapid progressors never had that early immune containment. They had high levels of virus right from the beginning. So these are some of, there's probably other reasons for it, but this is some of the reasoning for the difference between the rapid and slow progressors. Now, not only did I work for the government, but during these years I was married. And at one point, I don't remember exactly when, my wife said to me, "You are the answer to my prayers." I thought that was nice. But then she said, "You're not what I prayed for, but apparently you are the answer." So I always like that thought. I want to talk a little bit about hepatocellular carcinoma. This was a study we did with Dr. Yasuo Tanaka and Dr. Mizukami in Japan. They were experts in molecular clocking. You could take the virus at two different points of time and extrapolate back to when a particular genotype diverged from some common ancestor, and then when it spread rapidly in the population. So if you look at the red, in Japan, the major genotype was 1B. You can see it arose around 1880, and then it was quite quiet until 1930s and 1940s, when it began to spread, and now it's leveling off in the Japanese population. But why did it spread in the 1930s, 1940s? Japan was at war. They were at war with China first, and then with the Allies. We learned in retrospect that the Japanese soldiers were shooting up with amphetamines and sharing needles. And this is why a huge proportion of the Japanese population, male population, got hepatitis. But now those people have died off in Japan, and they're not using needles anymore. In contrast, in the US, the virus genotype 1A arose around 1910, was quiet until the 1960s, 70s, 80s, when our own needle sharing epidemic began. And unfortunately, ours is not abating. We still have a lot of needle sharing, a lot of potential hepatitis occurring. The reason I show this slide is that there's a 30 to 40-year difference between these two curves. And that's important because Japan has an 8 to 10-fold higher rate of hepatocellular carcinoma than we do in the US. And they've always said that when you have the virus, as long as we've had the virus, you'll have the same rates. And indeed, that has proven to be the case. I can't show you this slide in time, but we've seen a threefold increase in hepatocellular carcinoma in the last decades. And it was said that unless there was a change in the standard of care, that by 2029, there would be a fourfold rise in liver cirrhosis and liver cancer in the US than what it was in 2009. Whoop, sorry, go back. Well, I won't go back. I do want to go back. How do I go back? Can you go back? Yeah, there you go. Next slide. So there was a major, dramatic change in the standard of care. So now we have these remarkable HCV-specific antiviral drugs, so-called direct antivirals, that hit different enzymatic targets that keep the virus from replicating. Go back again, please. And so we can now cure 95 to 100% of patients. It's really closer to 100%. And I'll get into what dilemmas that raises. But we now have the means to cure hepatitis C, not just keep it quiescent like with B or HIV, but actually cure it. So that's the end of the beginning. So we now have the discovery of non-A, non-B, the eradication of post-transfusion hepatitis, the development of these remarkable drugs and the ability to cure the virus. So one can ask then, could this be the beginning of the end of hepatitis C? Well, there's reasons to think that it could be, because there was no non-human reservoir for hepatitis C. So we don't have to worry about mosquito vectors. We don't have to worry about a bat coming out of a cave and biting an animal being eaten, and we get the infection. It's only human-to-human transmission. We had these very sensitive diagnostic tests. Now, point-of-care tests for anti-HCV and even point-of-care tests for PCR. And we had these curative treatments. So we have everything we need to cure this disease. And indeed, it can be cured. As shown by Egypt. Egypt has been the poster child for hepatitis C eradication. Egypt tested 60 million people, almost their entire population, because they had very high rates of hepatitis C due to a schistosomiasis vaccine that was contaminated that they gave out in the 1940s or so. So they tested 60 million people. They found about 5% had antibody, and then they did PCR, and they found about 4% had HCV RNA. They treated 2 million plus patients over the course of 4 years. They cured over 2 million patients, almost everybody, 98.6%, because they got around the US patent for the drugs, which now cost about $24,000 in the US, used to cost $84,000. They made it for $3 and sold it to the government for $10. The government said, well, we can afford that. And they got cheap testing as well. So the whole thing costs roughly $75, $100 US to set up the infrastructure, to do the testing, to treat people, to cure. They have only about 350,000 people still to test. And Egypt now is willing to provide these cheap drugs and cheap assays to the rest of Africa. So we're engaged now in trying to eliminate hepatitis C in Africa. The developed world is trying to do that as well. We're not doing very well here in the US, actually. We don't have a program to test everybody. So this is the triangle of elimination. We have to do this massive screening with rapid assays. We need to deliver the drugs, ideally, at the time you get the test results. So the ideal would be a finger stick. You get the test, 15 minutes later, you have the result, you give the drugs. The drugs are perfectly safe. There's almost no side effects. It's incredible. So you don't even have to follow the patients very much. So what we need now is the political, corporate, and really the moral will to make it happen. That happened in Egypt. It should happen everywhere. And this drug can be eliminated. This disease can be eliminated. However, it's not going to be on my watch. It's going to take some time, because I have a new philosophic perspective related to age, where I have age on the abscissa and give a damn on the ordinate. and this is me out here. So I'm trying, but this is you young guys. This is you. This is you young people in the audience. You have all this ahead of you. You have the potential to not only eliminate this disease, but do whatever disease you're interested in. You have a vast opportunity. It's the most exciting career you can have, to be involved in discovery, hopefully come up with something that's curable. It's an exciting life you're getting into, and I wish you all great success. So thank you. Thank you. Thank you.
Thank you so much, Dr. Alter, for that amazing presentation. I'm going to open it up to the audience if you guys have any questions.
Yeah, that's a very good question, a very difficult question. I don't think that has really been answered. My suspicion is that Win Arias started it all, but there is now a hepatitis C-like virus that has been found in dogs and in horses. So I said we don't have another reservoir, and it isn't an active reservoir, but it might have evolved from some animal. Most of these viruses have evolved from primates in Africa, and these primates are then killed in the wild. Their blood gets over people, they get eaten. So, but it has not been pinpointed as to how it arose. Other questions?
I believe we have a Zoom question.
Yes, for those on Zoom, just to repeat the question was about potential origin of the virus given the lack of a animal host. From Monty Krieger, he asks whether or not you've published the poem.
You know, I don't know. I think it's probably is published. We used to have symposiums every 3 years, hepatitis symposiums, and I did a poem for each of them. If there's time, I have another poem later if you want. I never had no Nobel dreams, but I think we're probably running late, so I won't do it. But so it may be in those 3-year things. My wife was wanting me to publish all my poems, but I haven't had I'm too far along the curve to do that.
Okay, for the sake of time, we're going to cut the question and answer short, but please feel free to talk to Dr. Harvey or Alter during the coffee break or during lunchtime. So let's go ahead and I'll introduce the Okay, I'll give a brief introduction to our next speaker. That's Dr. Charles Rice. Dr. Rice is a Maurice and Corinne Greenberg Professor at the Rockefeller University. He's one of the world's most accomplished virologists and a prominent figure in research on members of the Flaviviridae family, including hepatitis C. Among the many accolades that he has also won, and he has also won the Nobel Prize along with Dr. Alter in physiology and medicine. And the topic of his presentation today is a new model for studying Hepacivirus-associated liver cancer. Let's welcome Dr. Rice.
All right, can you hear me?
Yes, we can hear you, Dr. Rice.
You can hear me?
Sir.
Okay. Well, I don't know how come I always end up having to speak after Harvey. I mean, this is just, it's unfair. I mean, because, you know, how can you follow, you know, an inspirational talk like that? So that's, I guess, where I am again. So I'd like to sort of pick up on something that Harvey sort of mentioned, and that is sort of trying to understand more about how chronic hepatitis C infection sort of damages the liver, including promoting liver cancer. So I think we all know that this is an important topic, so I don't have to spend a lot of time sort of convincing you that this is something that we should be working on. It's, you know, the 6th sort of most common cancer in humans. And in terms of the mortality rate, it actually stacks up at number 2. So I think it's important. And there are various etiologies that you know: alcohol, hepatitis B, and hepatitis C. And as you can see in the United States, hepatitis C is still sort of a major driver still of liver cancer. In other parts of the world, HBV dominates. So Harvey told you about this sort of success story that, you know, spanned over 30 years for hepatitis C, leading to this remarkable cure that we have today that really can cure just about everybody, even those that fail an initial course of treatment respond to retreatment. So that number is probably in places where you can do this kind of treatment work. It's probably 99.9% cure. So, that's great. But there are some unfinished things, and we believe that a vaccine may be needed. And I think that the experience with SARS-CoV-2 certainly heightened the enthusiasm for developing new vaccines. And there's a lot of work in that area. And although, Harvey and his colleagues sort of showed that this chronic infection was associated with sort of liver disease progression, we still really don't know very well sort of what the sort of viral and host determinants are that actually drive the pathogenic process. And, we can't really sort of do these experiments, as Harvey said, in the chimpanzee. We can get samples, or at least we used to be able to get samples from patients to sort of look at this, but that doesn't provide an experimental model where you can actually sort of test hypotheses about your findings. So I think the good news about the era of DA therapy is shown here on the left, and I think everybody is probably not surprised by that. If you are successfully treated for hepatitis C with the current direct-acting antivirals, you're at lower risk of developing cancer, and early is probably better rather than waiting for more advanced disease. The bad news is that, of the sort of untreated established risk factors, which I went through a couple slides ago, cured HCV is a major risk factor for developing cancer. I mean, even when the virus is gone, some patients still develop hepatocellular carcinoma associated with HCV. Also true in the case of suppressed HBV. So that's the bad news, is that, sort of even with these great drugs to get rid of the virus, some pathogenic processes may have been triggered during chronic infection that sort of live beyond the sort of virus and can lead to cancer. So we have spent a lot of time trying to develop a small animal model where we could study some of these pathogenic processes. And one of the approaches was to try and adapt the virus to sort of murine hepatocytes. That hasn't sort of worked very well. The other one was to try and figure out, sort of what are the human specific factors that this virus needs in order to enter cells and replicate. And we spent a lot of time figuring those out and making transgenic animals, but they were infectable with HCV, but it was a low-level infection that was resolved and didn't establish chronic infection. And then the third option was, sort of, maybe just sort of look out there in nature and see if we could find a related virus in a natural host that would allow us to begin working in this area. So that's what I'm going to talk to you about today. The bottom line and the sort of conclusion at this point in the talk is that we were never able to, despite all these different attempts, really create a physiologically relevant small animal model, a mouse model. So as Harvey said, sort of with the dawn of next generation sequencing, we began to see that there were lots of viruses out there rather than the very tiny subset that we had focused on because they cause disease in humans, animals, or plants. And so I was fortunate in the sense that I had a friend at the Mailman School of Public Health, Ian Lipkin, who sort of falls into the category of a virus hunter. And as Harvey mentioned, one of the viruses that was first found in animals actually was in a sample from dogs, but it actually turned out that it wasn't a canine Hepacivirus, it was actually an equine Hepacivirus. So I don't know how that happened, but anyway, so we actually, Ian Lipkin gave me a call and said that they discovered this. We started working on this with a group at Cornell. And actually, sort of this is not the small animal model that we wanted to work on. And really the Rockefeller campus is very nice, but I think we just didn't have the ability to house horses on campus here in Manhattan. So the next call from Hien came, and they'd actually found a HCV-related virus in deer mice. And that virus could establish chronic infection in deer mice, but it turns out that deer mice are so phylogenetically different from laboratory mice that basically none of the advantages of having a laboratory mouse model would have been possible with deer mice, because we just had to reinvent all of the reagents. Then there were Hepaciviruses found in bats. Again, not an easy sort of animal model to set up, and also in gibbons. But the real breakthrough came when Hien teamed up with some pest control companies here in Manhattan and trapped a bunch of Norway rats and basically started looking at their tissues by sort of deep sequencing. And so here's a picture of some of these rats. I apologize for those of you that are rat adverse, but anyway, it turned out that we got this virus from Hien, and Eva Billerbeck in the lab showed that the rat virus could actually infect laboratory mice. So this was, a rodent model, either in rats or in laboratory mice, that would allow us to begin to look at the impact of Hepacivirus infection on some of these disease processes. So we were very excited about that. Sort of Eva went on to sort of characterize this in some detail. What we found was that you see in this graph here a rapid rise sort of in viremia, and then typically the virus is cleared in, say, C57 black 6 mice in 2 to 3 weeks. There is associated liver damage, but basically it's an acute resolving infection. And this just shows you some work looking at immune cell subsets sort of in the liver after infection. And you can see that NK cells are sort of showing up in higher frequency in the liver, CD4 T cells, particularly in the liver, sort of later on during infection, and that sort of correlates with clearance of the virus. Now, it wasn't a chronic infection, but using a trick that the LCMV folks use to promote chronic infection, if you deplete CD8 cells, you can see in this graph in gray, we actually see a lag in the clearance of the virus. But when the CD8s come back after this depletion, the virus is cleared. But if you deplete CD4 T cells, then the virus becomes chronic in C57 black 6 mice. So that was very exciting. And Eva sort of went on and showed that this chronic infection persists after the CD4s come back. That happens after several weeks. And we could maintain these chronically infected mice for months sort of in the laboratory. And so this just sort of summarizes the features of this that Eva discussed, that she published in her paper in 2017. So it's a fully immunocompetent mouse model. We believe that the infection is exclusively hepatotropic, because if you take a miR-122 knockout mouse, and this is a microRNA that's required by HCV for replication, if you knock that out in the liver, the virus, we can't detect any active replication of the virus. So it's probably a hepatocyte-specific virus. There is the induction of a strong intrahepatic immune response, immune-mediated liver damage, T-cell-dependent clearance, susceptibility to reinfection, and T-cell exhaustion during chronic infection. So many things that are similar to what has been seen in chronic HCV. So we think that this would be a good model to really study pathogenesis, and also potentially to test sort of vaccine platforms, at least, to see how they work in this model. So what I'm going to tell you now is sort of all unpublished stuff. But it turned out that despite my pleas to people in the lab to sort of cull any animals that are actually not actively being used for research to try and maintain the sort of lab budget in the black. There were some ancient mice that had been chronically infected with NRHV for, I think, 18 to 24 months. And when these animals were sacrificed, the liver was a mess, and there was all kinds of sort of pathology. But we didn't really have any controls, and so we decided to start a pilot experiment to see if we looked at sort of longer times after chronic infection, if more severe liver disease would be apparent in these mice. So this was spearheaded by Mariana Batista, shown here on the left, and Juliano and Ana, who were visitors from Brazil, great scientists. Sean Chen, who's from Scott Lowe's lab, but now a postdoc with us, and Tyler Louie, and it's a multi-institutional and collaboration with Brad Rosenberg and Alice at Mount Sinai, and Tim Sheehan at UNC, and Scott at Sloan Kettering. So anyway, here was the sort of pilot that we set up, about 100 mice, sort of split between males and females, and an uninfected group. And CD4 depletion, basically infect the mice or sham infect the mice. And then the plan was to sort of take serum samples periodically throughout infection, but also have terminal time points at 6, 12, and 18 months. And so we initiated this experiment, and it turned out that at about the 6-month time point, New York City became sort of the epicenter of COVID in the United States. And so that really sort of complicated what we had planned in terms of harvesting and analyzing all these tissues. But anyway, sort of we carried on. And here you can see what we saw in terms of viremia. Viremia is very high early on, and then it sort of reaches a lower set point, presumably by exertion of some level of immune control. We don't see any difference between males and females there. And then if you sort of look at sort of the rare occurrence of animals that actually clear the infection after this transient CD4 depletion, there's only a couple of the males that actually do that, and more of the females. So there may be a difference in sort of long-term clearance rate. So one of the things that we are looking at are sex-dependent differences. I won't talk about those today, but there do seem to be some, as there are in HCC associated with chronic HCV in humans. So we do see evidence of progressive liver damage. This just shows you some sort of fibrosis sort of developing. We see these lymphoid aggregates, which are also often found in patients with chronic HCV. And there's a correlation between basically scores of inflammation relative to fibrosis. You can see this at the 7-month time point, and also at 12 months. These open symbols are basically the controls. This starts to break down a bit at 18 months because these mice are getting old, and we do see some evidence of sort of liver pathology in a fraction of them, even at these late time points. So again, really sort of no significant difference here due to sex, and we do see sort of progression. We don't see the development of cirrhosis in this model, unlike what we see in humans. But I'm not sure this is sort of a characteristic of mouse models in general. So then there was a surprising observation, and these are some livers from animals sacrificed, I think, at the 18-month time point. These are from the sort of control shown at the top, and basically the sort of chronic NRHV livers basically showed these liver tumors that we have begun to characterize. And it turns out that the penetrance of this in the surviving chronically infected males is almost 100%. All of them sort of develop liver cancer. So it's higher than what we see in humans, obviously by an order of magnitude or more, but nonetheless, I think that we are looking at an inbred animal population in this model. So I wouldn't expect it to to behave like an outbred complicated model like people. For the females, at least in this pilot experiment, they were less likely to develop these sort of liver tumors. And there were some slight differences in tumor volume. This doesn't really seem to be holding up between males and females in a second pilot experiment that we're sort of in the process of analyzing right now. So we see some of the hallmarks of the tumor tissue from these animals, and I won't take the time to sort of go through, but these do look like sort of well-differentiated hepatocyte things. They're actually very heterogeneous. The inflammation is quite variable, as it is in hepatitis C. And again, we do see these lymphoid follicles that are something that people are interested in because they seem to, if you find these in tumor tissue, they seem to sort of correlate with clinical results on some of these checkpoint blockade inhibitors. So the gene expression patterns in these tumors, animals sort of are very similar to what we see in people. So we think that this is at least sharing some of the characteristics of HCV-associated HCC. And we've done a little bit of genetics on these. Actually, 3 of these mice shown on the left have mutations associated with KRAS. This is a common driver in some cancers, not so much in liver cancer, but we do see it occasionally in HCC. And if you look at sort of markers of transcriptional markers that have been characteristic of HCV-associated HCC, you can see that some of these mice actually share those markers. So I'm not showing a lot of data here, but I just wanted to sort of throw this out to the group because we're really excited about this model. We have a chronic Hepacivirus infection model that sort of leads to hepatitis, fibrosis, and cancer. There may be some differences between males and females, so we're setting up to study that. These spontaneous associated HCCs are actually quite heterogeneous, so that'll be interesting to try and unravel. And there are common markers with human HCC. And there is a putative driver mutation, KRAS, in at least one class of these tumors. And so we think that this is really, maybe the physiologically relevant mouse model that we've wanted for all these years to explore pathogenesis and the nuances of HCC in a controlled environment. So it's one thing that I wanted to mention, and this is really interesting, is I don't know how many of you are familiar with the Collaborative Cross resource, but basically these are a group of genetically diverse inbred mouse lines. And so you have sort of a collection of these inbred lines that are genetically very different from each other. They include some laboratory strains like C57 black 6 mice. And you can use these to sort of interrogate a phenotype that you might be interested in and figure out the genetics that drive it. And so we have been doing that with Tim Sheehan at UNC. And basically what Tim has found is that, I won't go through the details here, but what Tim and Martin have found is that if you check a number of different collaborative cross founders, I'm just sort of showing you looking at sort of viremia in these plots over time. And this is a beautiful paper that Tim published in Hepatology about a month ago. The C57 black 6, sort of resolve the infection as I've shown before. And then the 046, 071, and the one in orange, but this is covered up by something on my screen, really have different rates of virus clearance. And in the case of CC071, these guys become chronically infected with this virus, sort of without any kind of immune intervention. So one can now use this resource to basically make an F1 and an F2 of C57 black 6 mice and CC071 to actually sort of map the loci that might be responsible for this sort of dramatic difference in resolution versus chronicity. And we're hoping to use this also for pathogenesis in cancer studies as well. So I won't go through this, but again, the CCO71s that become chronically infected sort of show these fibrosis signatures over time. And so some of the things that we're in the process of doing here to, sort of look at the effects of sort of pre-existing drivers. These can be sort of delivered to the sort of mouse liver in the absence or in the presence of Norway rat Hepacivirus infection, and we can sort of study how virus might be influencing the development of the tumor or the surveillance of the tumor. We're interested in sex and comorbidities in this model, role of fibrosis. One of the things that the Low Lab has done is to actually sort of explore the use of CAR T cells that sort of recognize markers of cellular senescence. And in some of their experiments, they've been able to show that that actually sort of stops the development of fibrosis. So we're interested in sort of trying that to see if it might influence the development of HCC in this model. We're also working out methods to actually cure the virus. And this sort of gets at what I, sort of mentioned at the beginning, which is, in the case of HCV, we know that there are patients where we get rid of the virus, usually ones with more advanced liver disease, and they still develop hepatocellular carcinoma. So we need better diagnostics to sort of figure out who's at the greatest risk, but we also need to know, I think, sort of how long do you need to be chronically infected before the die is cast and you're going to get cancer anyway, even if you get rid of the virus. And with this model, we can actually do a time course and test that. And immune function dysfunction on the pathogenesis in response to immunotherapy. So this I think is going to be interesting. We already know in this model that chronic infection is associated with basically a sort of a stunned phenotype sort of in the T cells. So we're going to look at that. And then, as I mentioned at the end, sort of the role of host genetics in infection outcome using the collaborative cross sort of resource. So that's it. I'm sorry if I went too long. But anyway, there are a lot of people that are involved in this at various stages over about the last sort of decade now. And the cancer angle, though, is all sort of new unpublished data and sort of driven, as I said, by Mariana Batista, a really talented postdoc in the lab from Brazil, and many others. Okay.
Okay, thank you, Dr. Rice. Okay, so we have time for one short question before we transition.
People want that coffee break. Uh-oh, don't give Win the microphone.
Whoops, Charlie, thank you very much for being with us.
Oh,
so there's a great deal of interest at the moment in the role of the microbiome in influencing human HCC and attempts to produce it in animals. Is there any information as to what the microbiome may or may not be doing in the Norway rat model?
I think it's a fantastic question. Maybe we should get together with, Barbara Ehrman or somebody and, sort of take a look at that. I mean, we do have people that, sort of do microbiome work sort of here at Rockefeller. Right now, we're just kind of in a struggle to try and get some funding to, I didn't sort of realize that if you start doing these long-term cancer experiments, that your animal bill goes up by somewhere between 10 and 20-fold. So I think that we'd love to do your experiments, but I guess we'll have to come begging to NIH to see if we can get some money to do them, but it'd be fascinating. I mean, I'm sure the microbiome is playing a role in this. We haven't even looked at, sort of what happens with this virus in, say, notobiotic mice, or ones that have been reconstituted with, sort of more defined microbiota. So that's a whole area that is ripe for somebody to tackle. Thank you for the question.
Thank you so much for this fabulous talk. This is John Gisabo. Great to see you, Chuck. Can you hear me?
Everybody wants the coffee break, I think.
Okay. So Charlie, it was wonderful to hear you. This is John Gisabo. I just wanted to ask if you can comment on microRNA 122 in these mice. You mentioned that obviously that's a host factor for at least Hep CV, but in hepatocellular cancer, typically miR-122 goes down. So I'm just curious.
Yeah, well, yeah, what you're alluding to is if you sort of knock it out, the sort of knockout mice, will develop liver cancer with miR-122. So, yeah, I don't know, sort of in vivo, sort of if the virus is really sort of sponging enough of the miR-122 for that to actually be, sort of playing a role. I mean, there is some fascinating work that is being done by a former trainee of Jens Bucht, and then who did his postdoc here in my lab at Rockefeller, who is actually very interested in this and probing it with the sort of NRHB model. And they have managed to actually sort of make a replication competent sort of version of NRHB that does not require, it appears not to require miR-122. So it'd be very interesting to sort of compare the behavior of that versus, one that does require and could potentially sponge miR-122 and sort of mimic, what happens in a knockdown or knockout environment. But yeah, we haven't the only mice that we've examined are the knockout mice, and these were short-term experiments just to see if we inoculated them with NRHB, if we got any virus back. But I think in terms of the sort of the pathogenesis, it's something to keep in mind, and it's probably something that dampened the enthusiasm of the drug developers that were, sort of targeting miR-122 with ASOs. Although, if you treat somebody for with an ASO for 2 or 3 months or something like that, it's not like, sort of being knocked out throughout development. So anyway, I don't know if that answers your question. I could ramble on for a while.
Thank you.
Let's go ahead and give Dr. Rice another round of applause.
Okay, everybody, enjoy your day.
Thank you. So we are aware that we're running a bit of time. And so I'd like to transition to the next speaker because she's also in Zoom. And then after that, we can take a coffee break. So let me go ahead and introduce Dr. Hien Dang. She is currently a tenure-track assistant professor at Thomas Jefferson University with the Department of Surgery at the Sidney Kimmel Cancer Center. Her lab studies the oncogenic role of RNA binding proteins in primary liver cancer using multi-omic approaches. And in addition to bench science, her team is currently collaborating with medical oncologists and surgeons to develop biomarkers to identify patients at high risk for HCC. Her topic today is leveraging splicing to target HCC.
Good morning, everybody. I'm not sure if you can hear me. And I would like to share my screen.
Hang on 1 second. We can hear you.
We can hear you. Can you share your slides with us now?
It says host disabled participant screen
Fantastic.
sharing.
Okay, give me just a moment here. Okay, you should be a co-host now. We should be able to have you share slides.
Great.
Fantastic.
Cool. Okay, well, this is really hard. I'm super nervous and shaking because I'm like going right after two wonderful speakers and Nobel laureates. So don't mind me if I'm shaky. Anyways, I'd like to thank the organizers as well as the American Liver Foundation for their support and allowing me to participate via Zoom. So I can't move on. Let's see. Oh, there we go. So I'm not going to go into details about liver cancer as Dr. Rice has kind of already mentioned it. What I will emphasize is that liver cancer is one of those cancers that is on the rise in the next 5 to 10 years. It's projected to increase by 40%. And that's due to the increased rates of obesity or NASH, which is now known as MASH. My lab is mostly focused on HCC, the primary focus of the primary form of liver cancer. And a lot of this whole process occurs in about 30 to 40 years. And a lot of patients with cirrhosis do not end up with liver cancer. A majority of those patients actually die of end-stage liver failure, although a small percentage do end up with HCC. So one of the main focuses of the lab is essentially identifying novel drivers of HCC. And we focus on RNA binding proteins mostly because there's this concept that these proteins or RNA binding proteins can interact with multiple RNA species as well as multiple RNA targets. So the idea is that one deregulation of one RNA binding protein can modulate or affect multiple different types of RNAs, and that can then drive HCC transcriptomic changes, and that these changes are drivers of the disease or signatures of disease. And one way to really model or identify these drivers is by an integrated genomics approach, where we looked at 1,200 different HCC patients from various etiology. So it's hepatitis C, hepatitis B, and NASH or NAFLD at the time. And what we found was that there's about 60 RNA binding proteins that are altered at both the gene copy number and the mRNA level. And the rationale behind this is that if an RNA binding protein is important for cancer, or HCC in this case, then it must be selected during tumor evolution. So you should expect to see a high correlation between mRNA expression, which is the functional aspect, and then gene copy number alterations, so at the DNA level. And among those is one RNA binding protein known as negative elongation factor E, or NELF-E. And one of the things that we use as the litmus test to see whether this protein is worthwhile for studying is by seeing or investigating whether the expression of this protein or the gene copy number alterations in this case is associated with patient outcome. And this is 2 different independent cohorts. The Liver Cancer Initiative is mostly made of HBV-induced HCC, so a lot of these patients are Chinese. And then, of course, the TCGA LIHC cohort, which is American cohort, and there's a majority of HCV, HBV, as well as NASH, although there's only like 5 or 6 patients that are NASH. It's really hard, and I have to transition to MASH now. But and what we were able to observe was that when you have high levels of NELF-E at the mRNA as well as gene copy number, you see that there's a poor outcome for these patients. The other litmus test was whether this RNA binding protein is really important throughout the progression of the disease. So we looked at the gene expression of NELF-E at cystic nodules of specific patients, low-grade dysplastic nodules, high-grade dysplastic nodules, as well as early onset HCC. And this is the RNA level. And what we see is that NELF-E expression is slowly increased throughout tumor progression. And so based on this, we thought that NELF-E is quite important for HCC. And so what is NELF? NELF is a part of the NELF complex, and the complex is made up of 4 different proteins: NELF-A, B, NELF-C, and D is one protein, and then of course NELF-E. And NELF-E is actually the RNA binding protein that has an RNA recognition motif that directly interacts with nascent RNAs. And so the NELF complex directly interacts with POL. That's through NELF-A, and when it interacts with POL2, it induces POL2 pausing, which is essentially a pause of transcription. And this occurs after transcription is going through from 30 to 70 nucleotides downstream. And this pausing mechanism occurs in about 30% of the genes, and most of these genes are stress-induced genes. The hypothesis is that if the chromatin is already open, transcription is already engaged, but it's paused, then if there's a stress event or anything that requires like a heat shock protein, for instance, then it takes a lot less energy for for transcription to occur than it is to have to, do the whole thing where you have the pre-initiation complex bind and then the whole process begin again. NELF-E is also a part of, is an important protein that's important for processing of replication-dependent histone mRNAs, and it's been demonstrated to undergo condensation or phase separation to downregulate transcription. And in cancer, at least in HCC, and this has been reproduced in pancreatic cancer as well as gastric cancers, not by our lab, by other labs, we were able to show that when you knock down NELF-E in these HCC cell lines, I'm only showing you 2, but we've done this in 6, you see that there's a significant decrease in cell proliferation. We also see a significant loss in migration and invasion as well, but I'm only showing you limited data. In addition, we were also able to xenograft these cell lines into non-skinned mice. They're immunocompromised mice, and we can show that when you knock down NELF-E, tumors actually do not form. And we were able to demonstrate that NELF-E actually works through MYC, the proto-oncogene that is that is deregulated in 70% of all cancers, but is altered in more than 80% of HCCs in various different ways. At least in our model, NELF-E actually has an effect on how MYC binds to its promoter regions of its downstream targets. And here we're showing that when you lose NELF-E, you see a significant reduction in MYC binding at the promoter regions of 2 highly proliferative genes. And then we also collaborated with Wen Xu at UMass, and he had this beautiful model where he uses a transposase system or a sleeping beauty model, where he overexpressed NELF-E in these MYC-induced mice models for us. And we saw that NELF-E alone had no difference or no effect in tumor formation. But in the well-established MYC model, MYC overexpression was able to induce about 2 to 3 tumors. But when you have NELF-E and MYC together, you're able to at least 3-fold increase the number of tumor nodules found in these livers. And so NELF-E gene amplification or NELF-E overexpression is very important in driving HCC, and this is really more or less through MYC signaling. So one of the things, or one of the aspects of what my lab was trying to establish is was to ask whether is it just gene amplification or is there other genetic mutations that's really driving this phenotype that we're seeing as well. Because gene amplification is only seen in about 20% of our HCC cohorts, but we also see that there's a larger population where NELF-E is also overexpressed. So we set off to sequence the 90 or so HCC patients. These are all HCCs with HBV, and most of these patients are, actually all of these patients are Chinese, excuse me. And what we did was essentially just set up for Sanger sequencing modular domains, because the hypothesis at the time was that if you sequence the modular domains, those would be the most likely mutated regions because they're most functional. Although that may not always be true. But anyways, what we did do was sequence those, and we didn't actually find any mutations. So that was one of those days where it was kind of dark, but that's okay. So we then did find was this really unique SNP, or single nucleotide polymorphism, that was heterozygous and found in about 8% of our population. And this was a G to A polymorphism. And so again, we have to go back and see whether this mutation actually has a specific clinical outcome. And when we looked at it and looked at our liver cancer cohort of HPV HCC patients, we saw that patients with the SNP actually did a lot better compared to patients with NELF-E amplification or the wild type. And then, although it wasn't very significant with a Cox regression analysis, we did see that patients with the SNP did do far better than both the wild type as well as the NELF-E gene amplified cases. And these are all mutually exclusive. So you don't really have the SNP in patients where they're wild type, or excuse me, in the amplification cases. So we did more analysis, and this is work done by a surgical resident in the lab who was with us for 2 years. And he found that patients with a SNP were found in about 1% of the general population. But there were about 5% of them in Southeast Asians. This is using the genome SNP database. And then in our cohort, about 3% of HPV-HCC patients have the SNP, and it went from 8% to 3% because when I moved to TJU, we started working with all the clinicians and the medical oncologists and the pathologists. We were able to collect more samples and increase our cases to include both HCV as well as NASH. And we're like starting to look at adenoma HCC patients as well as pancreatic cancer patients or colon cancer patients, really any patients, just to see if we can find these SNP in these patients. And so that decreased from 8% to 3%. Then we also found that patients with SNP had a survival that was much longer. And then, of course, they were less likely to recur, or they recurred much later than the wild type patients. So what does this mean, and what does the SNP really do? So, and I came from a cancer biology and a bioinformatics background. So if you're doing splicing, please excuse me, because this is all new to me as well. So one of the things that most splicing scientists do is essentially take the loci with the SNP in it and test to see whether there's an effect on the gene expression or splicing. And so what we did was clone the loci, including exon 9, exon 10, and exon 11. And the SNP is found about 103 base pairs into between exon 9 and exon 10 or intron 9. And so we cloned this, and then we tagged the GFP, and then we did PCR just to see what happens to NELFI expression when you have the SNP compared to the wild type. As you see here in the gel, when you do have the SNP, there's a reduction in the NELFI expression, although this is just a PCR, so it's really hard to quantitate. So what we ended up doing is taking this whole thing and putting into a dual luciferase assay so that we can quantitate it. And we were able to also observe the same thing. And what we saw was, again, with the SNP, you see a significant reduction in the expression or the transcription of NELF-E wild type. And so we went back to the patient's samples just to see if we're able to observe a correlation as well. And when there were some samples that we were not able to extract RNA due to the poor quality as well as the age, a lot of these samples were from FFPE blocks, and so RNA is highly fragmented. But for the samples that we were able to get some quality in, we were able to see that it was with the SNP, you do see a reduction of this NELF-E expression. And so what I've said before is that NELF-E is, the isoform of NELF-E is found between exon 9 and 10, and it's pretty close to the polypyrimidine track, which is where most spliceosomes tend to kind of bind so that it can cause splicing. And so the next thing we wanted to test was whether there's a secondary isoform. So if it's potentially there, it might have an effect on splicing. Maybe it's not splicing out introns, and maybe there might be something else that, or in a secondary isoform that is happening. And so what we did is, if you notice here, we had an ATG, which forced it to undergo translation or surpass, go into transcription. Here, we removed the ATG just to force it to avoid nonsense mediated decay. And when we did this analysis, we transfected into the cells, and we found that when we removed the ATG, you can see that there's a secondary isoform. And we sent both bands for sequencing, and what we found was that the second isoform had a lack of, or a loss of, exon 10. So there was an exon 10 exclusion when you have the SNP compared to the wild type. And a lot of this work is done by the talented grad student, Laura Reynolds. If you can believe it, she's a second-year grad student, super smart. So one of the things that the field the splicing field does is utilizes antisense oligos. Dr. Rice was mentioning this before, the MIR 122 studies. And these antisense oligos essentially sterically bind to splicing sites, at least in our case, bind to the 3' splice site as well as the 5' splice sites to sterically bind to those regions and block splicing to occur. And that can then cause exon 10 exclusion. And so we designed two different ones, one here at the I9 and here at the E10. And we can show here, and we did this in multiple different HCC cell lines, just because of NELF-E's role in transcription of different virus. we had to do in different HCC cell lines just to make sure that there's not really an effect because of the viral transfection of different HCC cells. So we can see here that this actually did work. We again sent the sequencing, the different bands out for sequencing, and we can see that in this case, exon 10 exclusion did occur. So our ASOs really did work, and it worked really well. And this again was done in 4 to 5 different HCC cell lines. We did it in like 3 different pancreatic cancer cell lines too, just to make sure that what we're seeing is reproducible. And so the next thing that we did was ask, can we use this to now target NALF-E in HCCs? And so we performed cell proliferation, migration, and invasion assays. What I'm showing you is only proliferation assays due to time. And what we found is that when we knock down or use these ASOs to induce the second form generation, we see a significant reduction of proliferation. We also see a significant reduction of migration as well as invasion. So we think that these ASOs are really good at knocking down the wild type. In terms of what the second isoform here does, we don't really know. We're in the midst of trying to figure out how stable this isoform is and whether this actually becomes a protein. We ran a Western blot also, and we can see here that there's a reduction in NALF-E wild type, but we also see this really nice second band. But we don't know for sure what the second band is and whether the second band is really a product of this isoform. So currently, Laura is doing those studies. So basically what we saw was that these ASOs targeting NALF-E was really good. So we wanted to see whether we can do this in vivo. So we started a collaboration with interventional radiology, who has this beautiful system of enhanced ultrasound. And one of the things that we normally do in the labs is making these xenografts where we'd actually have to do surgeries. And as a scientist, I am the worst surgeon in my, and I don't really have a very good rate in making sure that my mice are alive after surgeries. And so this was like the best way, at least for me, to inject or generate xenograph models where we use ultrasound to look for the liver and then inject liver cancer cells into the liver. And this is what you see, which is the needle. We have a live video, but for the sake of time, this is probably the best. So you can see here's the liver, here's the needle, and we were able to inject into the left lobe. And so what we did was also tag these HCC cells, and in this case, we injected 1 million HUH1 cells. And within 2 weeks or a week and a half, we already started seeing nodules. And at the point of nodules that we were visibly able to see, we started treatment at 25 mg per kg for each mice. And here, this is a preliminary study. We did about 4 mice for each group. And what you see is like right here is where we started treatment. And the mice that had no treatment, the tumors progressed and we had to sock them due to tumor burden. And then 4 weeks post-injection and 6 or 8 doses after, doses later, we saw that these ASOs was actually having a really nice effect. And so we were able to decrease tumor burden significantly. So currently what we're doing is increasing our N and also testing to see whether if the tumors are much bigger, can we also target NALF-E when the tumors are much bigger. In this case, they were pretty small, which is more representative tumor resectable HCC. When the tumor is a little bigger, more localized throughout the 2 different lobes, perhaps that would be more likely of a case to mimic the advanced HCC cases. So that would be an instance where we would do treatment to see whether it would work. The other thing that we're also looking at is using microbubbles, which are these like tiny nanopores that you can then burst using ultrasound. So we would deliver the ASOs into these mice and only burst the bubbles that are found near the tumors using enhanced imaging. That way we would get treatment directly at the tumor as opposed to outside of the tumors, because during these studies we found that there's a little bit of toxicity. So in summary, what we've been able to show is that NALF-E is really important in HCC, and it does this tumor promotion through mixed signaling. And we identify this novel SNP that's enriched in Southeast Asians. It's found in about 5% of Southeast Asians compared to the general population, which is less than 1%. And we also found that HCC patients with the SNP were less likely to recur, and they do tend to have better outcomes compared to wild type as well as NALF-E amplified HCC cases. We then also identified that the SNP promotes exon 10 skipping, and this then causes an out-of-frame shift, and that can have an effect on NALF-E mRNA expression. And more importantly, we were able to demonstrate that the ASO, at least the I9 ASO, was able to target NALF-E and reduce tumor burden in vivo. And just to mention that because it's right by the polypyrimidine track, we're also doing mechanistic studies to test whether this has an effect on spliceosome binding. So we're doing a little bit of that, trying to kind of dissect out the mechanism. And then, of course, the protein, we're going to do a lot of different assays just to figure out whether this protein is really the product of this isoform and whether this protein itself has a protective function, as we see here. And then with that, I'd like to thank everybody for coming and the opportunity from the American Liver Foundation. I was the ALF scholar in 2019, and I have to say, it was this award that really helped me feel a little bit more safe during really unsafe COVID times. And then, of course, I'd like to thank my current funding mechanisms with the Innate Cancer Center, and then our Biden Moonshot Scholar grant that we just received back in July. With that, I'll take any questions. Thank you.
We'll have time for 2 short questions. Oh, okay. I'll ask a question. I may have missed this, Dr. Dang, but when you treated with the ASO, you treated early on when you injected the HCC cells. Have you done studies or experiments where you've treated these mice after the tumor has already formed?
Yeah, we did. We did a preliminary study just to make sure that to test toxicity as well, and that was done, I think, in this. What year are we in? We're 2023. Sorry, I have a 1-year-old at home, and they don't sleep. So we did do that, I think, a few months ago to test toxicity, and we found that when the tumors are much more progressed, we weren't really seeing an effect. And we didn't know if we weren't seeing an effect because the tumors are already so far progressed, or whether the ASO actually got to the tumor. So we're currently trying to optimize the conditions so that the actual ASOs will get to the liver and do what it needs to do. So yeah, there was concern that the high concentration of the ASOs was causing toxicity. And so we need to try to figure out what the optimal dose still, but yeah.
Hello, everyone. Welcome back to our second session. We will briefly introduce ourselves. I'm Enrika Wedig. I'm an MD candidate at Wolfram Goessling's lab.
I'm Tina Lopez. I'm a grad student in the Knaus lab.
And we will guide you through our second session today. Please scan the QR code on this slide to view the whole program, and we're running a little bit behind, so we will go a little bit into the lunch break. I have the pleasure to introduce our first speaker, Dr. Hoodless. She is here from University of British Columbia. She completed her PhD at the Queen's University in Kingston, Canada, followed by her postdoctoral research with Dr. James E. Darnell at Rockefeller University in New York. She focused there on liver gene regulation. Dr. Hoodless serves as the director and distinguished scientist at the Terry Fox Laboratory at the BC Cancer Research Center. And her research focuses on transcriptional networks and their role in regulating liver and heart valve formation. Her topic today is gene regulatory dynamics in hepatic specification. Let's give it up for Dr. Hoodless.
Turned on. Oh, that's me. So I'd like to thank the organizers for inviting me. This has been it's been a fun week. I went to the AASLD first, my first time there. And so, but this is the highlight of my week. So this is great. Thank you. So the slide here that I'm showing is basically, there's my point. This is a section through a mouse 9.75 liver bud. So the liver starts to form at about embryonic day 8.5, about a day earlier than this stage here. And you can see that these cells here, I've lost my pointer, but that's okay. The green cells are marked by an AFP-GFP marker. And so those are the hepatoblasts. Those are the ones that are going to go on to differentiate, the bipotential cells that are going to go on to differentiate to the hepatocytes and the cholangiocytes. But what I like about this slide is you can see that these cells are really closely associated with mesenchymal cells, shown sort of down here in the purple, and the red cells, which are the endothelial cells. And even at this early stage, my lab's been really interested in trying to understand the cell diversity in this liver bud at this stage. But even at this stage, you start to see differences in the mesenchyme that are there, and as well as already those endothelial cells, they're starting to express sinusoidal endothelial markers. So there's a lot of heterogeneity in cell diversity, even within this early stage. And we do know that signaling from the mesenchyme and the endothelial cells are really critical for these cells to form. That all said, I'm going to move forward and say that today, I'm not going to talk about our mouse work. I'm going to talk about what we've been doing recently using human pluripotent stem cells to model development. And we've, as was said in my introduction, we're really interested in gene regulation. And so we've taken sort of an epigenetic approach to it. Whoops, there we go, back one. So just for a really quick introduction, I know epigenetics is become sort of a favorite topic, I think, ever since the genomics has really become and grown. But really, it's pretty simple. You know, the diversity of different cell types is primarily regulated at the level of gene expression and not the genetic code. We know that cells that are genes that are, there's my pointer. Genes that are not going to be expressed in that cell type, they get packaged away in this very dense chromatin so that they're not expressed, and genes that are expressed are through this more open chromatin so that they're able to be accessed by the transcriptional factors. One of the interesting things that's happening is that we can actually use epigenetic marks, so either histone modifications or DNA methylation, to really try and understand the structure of the genes and how they're of the chromatin, the structure of the chromatin and how those genes are regulated. So we have active marks, which constitute things like this is histone 3, lysine 4 trimethylation, and histone 3, lysine 4 monomethylation, which mark promoters and enhancers. K27 or lysine 27 acetylation, which marks whether they're active or not. And I'm not going to talk about DNA methylation today, but hydroxymethylation. There's also K27 trimethylation, which tends to be a repressive mark. So we really wanted to take a really unbiased look at how at hepatic specification. And I'm obviously not pointing this in the right place. There it is. And so we chose to try to use the human pluripotent stem cells. And there's several reasons for this. One of them is because you can actually get large numbers of cells. As you saw that mouse liver, it's pretty tiny. You can get larger number of cells. You can also get very defined stages. And so you don't have to sort the cells. And so you can get very defined stages. So the first thing, of course, we do is we take our pluripotent stem cells, we differentiate them to a stage called mesendoderm. And then the mesendoderm is then differentiated into definitive endoderm. So definitive endoderm is where the layer where all those hepatoblasts are going to come from. From the endoderm, we can specify, or we can go change it to a hepatic endoderm, which is really where the specification is taking place, and finally to hepatoblast. And so we took these different stages, specifically these 4 stages, and we actually generated ChIP-seq to all of the different histone modifications using histone 3 lysine methylation, 27 acetylation, and also 27 methylation. And then we classified the entire regions based on their histone marks into promoters or enhancers. They're a promoter if they have H3K4ME3, whereas an enhancer where they have H3K4ME1 but no ME3. And then active, whether they're marked with the K27 acetylation, and or repressed if they're also marked with the K27 trimethylation. And so when we took all of that data and we made these, basically we made these alluvial plots, and these alluvial plots contain a lot of information. First, we've divided it into promoters and enhancers. And on the promoter side here, you can see that there's about 16,000 promoters that are in this plot versus about 600,000 enhancers, which are included in this plot. And we have our different stages across the bottom: mesendoderm, definitive endoderm, hepatic endoderm, and hepatoblast. And each one of these lines is a different promoter or a different enhancer. And each one was classified as whether it's quiescent, so it had no histone marks, whether it was repressed, so it's got the K27ME3, the primed, where it's got the H3K4ME1, or active, where it's got the also the acetylation. And the first thing that you can see is that the promoters are much more stable than the enhancers. The enhancers are really highly dynamically regulated. And this is really, we believe that the enhancer regulation is actually really more associated with the cell identity almost than the promoters. The promoters tend to be active quite early. So you can see even by the definitive endoderm, quite a large proportion of these promoters are already in an active state. So if we focus a little bit further down onto the enhancers, I've just highlighted, there's a lot of stuff going on here, but I've just highlighted a few groups. And one of these groups here are ones that are quiescent in the mesendoderm. They become big, there's a big swath of priming that happens at that definitive endoderm stage. And then 97% of those primed enhancers are going to become, they never become activated. They either lose their priming, or they become repressed, or they just maintain their H3K4ME1, their prime state. They never become active. But there are a few that we can identify where we can see where, so this is kind of hard, where we can see that these primed enhancers become active in our hepatoblast stage. And even though it looks like a small number, it's actually just under 3,700 enhancers. So that's quite significant. And we can also see there's another line, which I've highlighted here, where these are enhancers which go from quiescent to active at the hepatoblast stage. And that's about 7,200 enhancers. So we call them primed to active or quiescent to active enhancers. We do believe that these enhancers are actually our liver. They're regulating liver genes. You can do the gene ontology and see that the genes that are associated with these enhancers are all sort of liver genes that you might expect. But you can also look here. This is using published data. If we take our hepatoblast data and we compare it to a data set for pancreatic progenitors, as well as lung progenitors, we can see that these enhancers, our primed to active enhancers, are active in our hepatoblasts, but not in the pancreas or the lung. So these really are marking liver enhancers that are required. This is again taking those primed to active enhancers and then looking at the promoters that are associated with those enhancers. And you can see that again, by the definitive endoderm stage, a large percentage of those primed to active enhancers, the genes that those primed and active enhancers are associated with, are already in an active state, even though the enhancer is not active. And you can see, again, if we take our pancreatic and our lung samples, you can see that the promoters of those genes, they're all active in all of those lineages. Whereas again, this is the prime to active. This is just the one that I showed before. You can see that it is specific for the prime, for the actual enhancers. You can see that it's prime. It's associated with hepatoblasts. So really that's telling us that those enhancers are really critical to defining the states. And you can, over on the, sorry, that pointer is just kind of wandering all over. Over here, you can see that these are the genes that are associated with those enhancers, and you can see there's the promoter activation occurring at the definitive endoderm stage, and this is the hepatoblast stage, and this is the gene expression. So the gene expression is turning on when the enhancer gets activated. So we really think these enhancers are important for this step. So the next thing we did is what binds to these enhancers. That's obviously the next question. And so we did just did some motif analysis to start that. And you can see this is the prime to active enhancers and the quiescent to active enhancers. And just for comparison, this is the definitive endoderm enhancers. And you can see that there at the definitive endoderm, you see all of the GATA, the FOXA, EOMEs, all of the candidates that you would expect. In our prime to active enhancers, we do see several factors. And I'm just going to focus on TBX3 and HNF4. Which are known to be function in hepatocyte liver development, but not really at these really early stages. So, and you can see they're both in that quiescent to active as well as the prime to active. In fact, TBX3 falls at the top here. So HNF4 is actually quite low, which is a little bit surprising on the list. But we wanted to follow up a little bit with, so to try to choose which factors of those lists we were going to follow up on, we basically turned to expression. And this just here shows the expression in the heat map shows the expression in our human hepatic differentiation cells. And you can see that, again, TBX3 and HNF4 are turning on at that hepatic endoderm stage. On the heat map on the right, you can see that this is our mouse data. And again, you can see at the bottom that TBX3 and HNF4 are turning on very specifically at that early endoderm stage. And this one's just for fun. This the one at the bottom is just a spatial atlas. This is now an old version of a spatial atlas. They're much more refined now, but this was one of the early ones. And you can see that there we go. That the liver bud, we got the pointer back. The liver bud here is very strongly expressing AFP, marking it for the liver bud, but it's also expressing TBX3. And it's very specific to TBX3. It's not expressed in those pancreatic regions. So we decided that we should go ahead and chip TBX3 and HNF4 at our different stages of differentiation of our pluripotent stem cells. And as you can see, we can see with TBX3, we see that at most the binding is actually occurring at the hepatic endoderm stage. With HNF4, we do see strong binding at the hepatic endoderm stage, but we also see some at the hepatoblast stage. It's much more evenly distributed. And so we were really interested in the TBX3. So we've gone on to look a little bit more in depth at that. Oh, so you have to double hit it and then go back one. Guess not. There we go. And so this is just a comparison. Basically, if we looked at those prime to active and quiescent to active enhancers, that we can see that TBX3 is actually bound to quite a large percentage of the enhancers. You know, about 80, looks like about 84% in the prime to active, as well as in the the quiescent to active enhancers over here. So here, this is a very messy diagram, but basically here we took our TBX3 again to look a little bit more at how TBX is actually functioning in these differentiation processes. We aligned up our TBX3 with the histone modifications. And again, as you can see, that the TBX3 is highly expressed in the hepatic endoderm stage here. And this is now we're looking at not just those prime to active and quiescent to active enhancers, we're looking at both promoters and enhancers. And just to show you that those are TBX sites, this is just the motif on the right here. We have the motif analysis to show that we are seeing a TBX binding site in the vast majority of those peaks that we see. So just to focus a little bit more closely, make it a little easier, on the promoters alone, you can see that they're promoters because they've got the H3K4ME3, they've got H3K4ME1 or stronger H3, and they've got H3K27 acetylation, as well as H3K27 trimethylation at various points. And I think the most surprising thing of this figure is that we see the majority of binding of TBX3 in a hepatic endoderm, and there's no H3K4ME1 at those sites in the hepatic endoderm. And there's also very little of the H3K4ME1 in those sites. And so, but all of those sites, you're now seeing strong promoter binding or histone modifications happening at the hepatocyte stage. So that's really different than a lot of transcription factors that we've looked at, like HNF4, SOX9, FOXA2, where when you see the binding, you see the histone modifications associated with the binding. And so we're not totally sure what this actually means, but basically that all promoters that are bound with TBX3 only acquire the H3K4ME3 at the later stages. And also the other thing that was interesting is that we're seeing a split 50-50 of these bivalent. So bivalent, we mean that they've got the H3K4ME1 as well as the K27 trimethylation, and active promoters where they've got the K27 acetylation. So if we, just to show you that it's not just an artifact per se, is that these are all those TBX3 promoters that I just showed you in the previous one. And these are all the other promoters that we can identify in our data. And you can see again that it's not just that the data is missing in our definitive endoderm and hepatic endoderm, that it really is a depletion of those marks at the TBX sites. And this just quantifies, on the right here, we just have pie chart quantifications of that, showing that the TBX3 bound promoters, they're either active, primed, or repressed. But you don't see any ones that are, or sorry, bivalent. You don't see any that are repressed per se. So we can do, the other thing that we did is that, again, maybe not super surprising, but if you now look at this content, the CpG island content of these promoters, if you look at all promoters versus the TBX bound, you can see there's a huge difference. And again, this has been noted before in that promoters that tend to be depleted in CpG islands are ones that tend to be more associated with lineage identity. And so it does seem to be required for TBX to bind that we need these low CpG island promoters. Just to show you quickly, in our last minute, is that so this basically you can look at the same analysis with enhancers. You don't have the strong H3K4ME3, which is not surprising. They're not promoters, but you still see that there's strong H3K4ME1, and you can see that there's this acetylation versus the K27 acetylation and the K27 trimethylation, showing that these ones are active and these ones are bivalent. And again, showing that it is specific. Back one, showing, so again, these are the enhancers at the top and then all of the other enhancers at the bottom. So it's not just that the data is lacking in certain areas. So this is basically where we're at. We think that we know from other studies that TBX3 can bind that complex, which will actually methylate the histones. And so we think that TBX3 is recruiting that complex to methylate those histone modifications. But it seems to me that as soon as those histones are modified, the TBX3 is getting booted out. It's being ejected. And it's possible that that ejection is due to the fact that they're by the histone modifications itself, or perhaps there's another transcription factor or a reader that's getting recruited in and it's getting booted out, and that those are recruiting the histone acetylation and the PRC2 complex to either give you the acetylation or the bivalent bivalent promoters. So basically, just in summary, we map the enhancers and promoters during hepatic specification and show that the enhancers are really more dynamic than the promoters. 97% of the enhancers that get primed in the definitive endodome do never get activated. And so those are thought to go on to be the other lineages, the pancreatic lineage, the lung lineage, that those enhancers are getting downregulated. We identified two groups of enhancers that become activated, both the prime to active and the quiescent to active, and also identified TBX3 and HF4 as potential regulators at these very early stages. And the expression data suggests that TBX3 plays a role in both mouse and human specification. And the next one, just TBX3 binds to these early hepatic specific enhancers. And we did look at pancreatic specific enhancers, and those are in those groups that get turned down. And TBX3 is not binding to pancreatic enhancers, even though it is thought to play a role in repressing pancreatic genes in the liver. TBX3 is recruited to promoters lacking CPG islands, and it mechanistically precedes H3K4ME1 or H3K4ME3. So we don't see TBX3 binding coincidentally with the H3K4 methylation. I don't believe it's a pioneer factor. It doesn't really fit. Because if you look at the data, you can see, if we look at other people's data, you can see GATA and FOXAPE binding before, so it's probably not a pioneer factor, and it requires those other pioneer factors to start the whole process. So it likely recruits those methylation transferases, and then it gets evicted by the actual methylation. So we think that TBX is really reinforcing that H3K4 ME1, that methylation, and then leaving. And the people, most of this work is done by Tabia, right here, with help from Jeremy, Sybil, and Becky, as well as some other members of my lab. And this work was actually done in collaboration with Stephen Duncan at the Medical University of South Carolina. And that's it. Thank you.
Thank you very much for sharing this story with us. And now we have some time for questions.
So Pam, very nice talk. I saw that there were like split domains. There were a couple of tracks where there were two TBX peaks, and then there were also the corresponding chromatin marks. Also, what kind of regions are that? Anything special about them?
I think those are basically two sites that are basically side by side. They're dimer sites. Although TBX3 itself doesn't dimerize, but I think that's what we're seeing there is in those regions. So those actually are very interesting, and we should definitely delve more into them to see what kind of genes are actually being regulated by those sites, because those could be some very.
Yeah, that was my question. Is anything special about those genes?
I honestly
that require the two domain binding. Yeah.
Yeah, I don't know. I don't know yet. We do have to look at that.
Let's give it up one more time. And if there's no more questions, transition to our next speaker. Actually.
Sorry? In the adult hepatocyte? So in the adult hepatocyte, TBX3, I believe, is quite low. It's not expressed at a very high level. I have looked sort of a little bit on those TCGA websites and stuff like that, and there is some upregulation of TBX3 in that you can see an upregulation of TBX3 on that site. So yeah. Sorry, I can't hear you. It's not on. Is it going up in HCC? Well, in some samples, it wasn't hugely overwhelming. It's not like GPC3 just totally like blows everything out of the water when you go on those sites. TBX3 is upregulated. We haven't done a clear study to sort of characterize it and actually quantify it or anything. It is, but you can see that it's upregulated if you go to the cancer browser and play around with it. I like to think everything embryonic is upregulated in HCC, even though I might be wrong.
Okay, if there's no more questions, let's give it up one more time.
All right. Hello? Oh. I talk loud. Hi, I'm excited to introduce our next speaker, Kendra Bence. She's the executive director of metabolism at Pfizer, where she leads a group focused on developing an understanding of the biological activities underlying metabolic dysfunction, with the goal of identifying novel ways to treat and eventually prevent metabolic disease. She has a strong commitment to training and mentoring the next generation of scientists, which has led her to run the Pfizer Global Postdoctoral Program. Today, she wants to tell us about her talk entitled "The Role of Phytine Inhibin E in Liver-Adipose Intra-Organ Communication." Thank you. Can you hear me okay?
Great. Much for that. And just to add my thanks to the organizers and the ALF. The Pfizer building is a 5-minute walk this way, and this meeting, I'm so glad we're here in person. This meeting has been great for myself attending in the past, as well as members of my team, and it's really just a great way to get to know the broader local liver community. So it's great to be here today. Just my disclosures: I'm an employee and shareholder of Pfizer, and my animal care and use statement. So metabolic, I don't know if we can get rid of that thing on the top there, so we can see the titles, but I'll just keep going here. So metabolic associated fatty liver disease is really just a description of the accumulation of fat or lipid in the liver, or hepatic steatosis, primarily in the context of obesity and type 2 diabetes and other kinds of metabolic dysfunction. Recently, many of us are struggling with the recent proposed name changes. It's hard to teach an old dog new tricks, so I will mostly go back and forth to NASH and NAFLD. Essentially, the distinction is that NAFLD was really meant to describe non-alcoholic liver disease, so the exclusion of alcohol as a criteria, whereas MAFLD is really meant to acknowledge the reality that a lot of fatty liver disease is really just driven by abnormal metabolic dysfunction. It's also important to mention, and probably this audience is very aware, but we still do not have an FDA-approved therapy for the treatment of NASH, which is the more severe form, non-alcoholic steatohepatitis, more severe form of the liver disease, which is characterized by inflammation and fibrosis. These later stages of the disease are characterized by fibrosis stage from F0 to F4, and you can see there's a huge increase in the mortality associated with this liver disease the further along in the fibrosis stage that you get. And it's not surprising that the projections for NASH over the next 10 or 15 years are increasing due to all of the increased rates of obesity and diabetes worldwide. And so one of the major drivers of fatty liver disease, get this thing to go forward here. I might have to go back one. There we go. So one of the major drivers of accumulation of fat in the liver is dysfunctional adipose tissue. And so in the context of normal healthy, chronic, intermittent caloric excess, what you have is a normal, healthy expansion of your adipose tissue shown here on the top. And along with that, you can get some new adipocytes differentiating and storing this increased fat that you're ingesting in a meal. Cell volumes of the adipocytes are normal and they remain insulin sensitive. However, in the context of really chronic excess caloric intake, what you get is a recruitment of macrophages to the adipose tissue, low-grade inflammation, reduced adipocyte differentiation, which limits your capacity to safely store this excess lipid. And importantly, in that state, you become insulin resistant and you have an increase in lipolysis and release of free fatty acids or spillover of fat into the circulation. And what this leads to is a buildup of adipose in places that you don't want it, like the liver and skeletal muscle, et cetera. And what we know is that not all adipose depots contribute equally to this process. So there's been many studies that have shown that the visceral depot is much more prone to changes in lipolysis and is a much bigger contributor to the overall fatty acid pool. And we've all heard of this pear-shaped, apple-shaped body phenotype, and this turns out to be really quite important in that when you have the pear-shaped fat distribution, where you store more adipose in places like your hips and your thighs compared to your abdominal area, this can actually lead to cardioprotection. So you have normal white adipose tissue function in this case. But shown here on the right with the apple-shaped distribution, you tend to accumulate more fat in the visceral compartment. This can lead to a disproportional increase of visceral adipose compared to your subcutaneous adipose, more adipose dysfunction, increased lipolysis, and insulin resistance. And importantly, this leads to a much higher risk of things like fatty liver disease, as well as cardiovascular risk. And clinically, this can be captured by a metric that we call waist to hip ratio adjusted for BMI. And this really accounts for the fact that you could have two people with an identical BMI, but a very different waist to hip adipose distribution. And what that means for that individual is that they could be much more prone to insulin resistance and fatty liver and diabetes. And so if you take this measurement and then look into the GWAS database and start to dig into the genetics a little bit, what's been shown is that when you have an elevated waist to hip ratio, your risk for type 2 diabetes, shown here on the right, goes up almost twofold. And we see a similar thing for cardiovascular disease, which I'm not showing here. But this leads us to this notion that potentially identifying targets or pathways that are linked to body fat distribution and adipose tissue health could actually be quite beneficial therapeutically in the potential treatment of things like fatty liver, obesity, and diabetes. And so our computational biologists at Pfizer dug into this. So they analyzed the exome sequencing data that's part of the UK Biobank. And what they had found, and subsequently others have found as well, is that there's quite a striking association with rare loss of function variants in the gene that encodes for inhibin E, also called activin E. I'll try and remember to call it inhibin E so I don't confuse you. But essentially, this is a hepatokine. And if you look in the green box there, you can see that the risk, or actually the waist-hip ratio adjusted for BMI on the top there is much lower when you have predicted loss of function mutations in this gene. Most of this is attributed to a reduction in waist circumference, a little bit of an increase in hip circumference, but primarily driven by a reduction in this visceral adiposity. It's been published as well in two really beautiful papers, one by Al-Nilam and one by Regeneron. And the important connection here is that not only does this gene, mutations in this gene seem to protect from visceral adiposity accumulation, but it also seems to protect humans from getting type 2 diabetes. So what does this do? So inhibin E is a member of the TGF-beta superfamily of activin ligands. These so inhibin is almost exclusively expressed in the liver, and within the liver, it's virtually exclusively expressed in hepatocytes. And because it is secreted from hepatocytes into circulation, we call it a hepatokine because it potentially could go on to act at distal peripheral organs. And the way these signal is that they either homo or heterodimerize, and they act on a family of receptors called the activin receptors, which are made up of type 1 and type 2 receptors. I'm not going to go through all the complexities here. This is a very complicated signaling pathway. There are numerous activins. There are numerous activin receptors. But what I'll just say is that the hypothesis, at the time we were working on this, is that inhibin E likely acts through some pairing of activin receptors to initiate SMAD phosphorylation and downstream changes in transcription. But at the time we were started to work on this, we weren't sure exactly what that coupling might look like. And also what intrigued us is that if we dug into 3 different cohorts in GO and looked at the expression of this gene in human liver under conditions of normal controls shown in red, NAFLD cases shown in green, or NASH livers shown in blue, it's significantly elevated in the context of NASH and NAFLD. And what was interesting to us about this is when we were looking through these sets of genes, this was the only one that was not associated with drug-induced liver injury, or DILI. So we don't think this is just a general marker of hepatic injury that is elevated. It seems to only be elevated in the context of increased lipid deposition in the liver and metabolic dysfunction. So we wanted to see if we could model this in mice, and so we looked across the circadian pattern here. I'm not going to use the pointer because I think that's what's messing up the advancing. So I'll just kind of describe what's shown here. So in the shaded bars, you can see on the bottom you have the dark cycle as well as the light cycle over the course of 24 plus hours in the fed and fasted state, and we do see it's correlative, but we do see an increase in inhibin E under conditions of which we have elevated plasma free fatty acid levels. So during the fasted state, your plasma NEFAs go up, hepatic triglyceride accumulates during the fasted state and then return once you've refed, and we see a similar trend for hepatic inhibin E levels. We also see similar results when we artificially induce an elevation in free fatty acids by injecting a beta-3 adrenergic agonist. And so we started, again, we were looking at these genes and exome sequencing data of UK Biobank that regulate waist-to-hip ratio. And we noticed that the receptor, activin type 1 receptor C, or ALK7 loss of function mutations in people, also had almost an identical association with waist-to-hip ratio. Again, being driven primarily by this reduction in visceral adiposity or waist circumference with a slight increase in hip circumference. And this was quite exciting to us because we already knew from the literature that ALK7 is a known regulator of lipolysis. It can regulate lipolysis with another activin ligand, activin C. And it's enriched in adipose tissue and has these very similar genetic associations. And so at that time, John Griffin, who's a very talented Pfizer postdoc in the lab, took on this really big project to try and understand and investigate this hypothesis that somehow metabolic dysfunction and accumulation of fat in the liver is leading to elevations in this inhibin E gene, which then can signal through the ALK7 receptor in adipose to suppress lipolysis in a sort of positive feedback loop and reduce fatty acid efflux to the liver. So John set about investigating this. The first thing he did was make some genetically modified animals. So for ALK7, he utilized CRISPR technology to make a K22R knock-in mouse of ALK7, which is a kinase deficient ALK7 model, as well as an I195T knock-in model, which is the exact amino acid switch that you see in the human genetics, both predicted to be loss of function. And then also just made inhibin E knockout mice, and he looked at their adipose tissue distribution. And, it's very exciting to see that actually if you use gonadal fat in a mouse as a proxy for visceral adipose, and the inguinal white adipose tissue in the mouse is a proxy for subcutaneous, you could do this gonadal to IWAT ratio and approximate waist-hip ratio that we see in humans in these mice. And we were really excited to see that, yes, indeed, in the presence of loss of function of either of the ligand or receptor here, you get a similar reduction in waist-to-hip ratio. So we then, one of the major ways you can reduce your visceral compartment is to increase lipolysis. And so we took a careful look at lipolysis. And again, under conditions of fasting, we do see for both ALK7 deficient animals and Inhibin E deficient animals an increase in circulating NIFAs in these mice. And on the two graphs on the right, if you treat with the CL ligand, which is this beta-3 adrenergic agonist, which will stimulate lipolysis, you can see that the mutant mice have a greater or enhanced response to this beta-3 adrenergic agonist. So we then wanted to move in vitro to really test this hypothesis more directly, that Inhibin E can directly suppress adipocyte lipolysis working through the ALK7 receptor. So we moved to immortalized mouse brown adipocytes and utilized, since Inhibin E is expressed into the media, we utilized a conditioned media system where we applied either empty vector conditioned media, mouse conditioned Inhibin E, or human conditioned Inhibin E to the cells, and showed that both mouse and human Inhibin E were capable of suppressing lipolysis in this system. And if you look at the graph on the right, we then employed two guides to CRISPR knockout ALK7 from these cells. And when we do that, so first, if you just looked at the first set of bars there, this is the empty vector. These cells were all stimulated with isoproterenol, so lipolysis is high. Activin C is our positive control here. So if you look at the white and the gray bars, which are controls, you see suppression of lipolysis as you'd expect. But if you knock out ALK7 with either of our two guides shown in orange and pink, you restore lipolysis. And again, we were quite excited to see that for both mouse and human Inhibin E, we were able to suppress lipolysis. And if you delete the ALK7 receptor, lipolysis is restored. And so, as I mentioned in the human genetics, this seems to be a loss of function gene where you get a more favorable adipose distribution and you're protected from insulin resistance and diabetes. So we were at first quite intrigued to see that when we looked at insulin sensitivity in our animals, we found the opposite. So these Inhibin E and ALK7 deficient mice, if you look in the center here at their insulin levels under the fasted state are much higher. And if you calculate HOMA-IR, which is a good measure of overall insulin resistance, it's elevated as well. If we subjected the Inhibin E knockouts to a more direct measure, an insulin tolerance test, where we inject insulin and look at glucose over time, you can see that the Inhibin E knockout mice shown here in red were pretty severely insulin resistant as well. Trying to move forward. I didn't even touch the pointer that time. Okay, so how can this be? Well, actually, it makes sense in a lot of ways. And that's because we know from numerous studies in people and rodents that if you elevate your free fatty acids and you chronically have increased lipolysis, these are central drivers of insulin resistance. So shown here on the left was a nice older study done by Jerry Schulman and colleagues showing that for a low glucose infusion rate, where you're more insulin resistant, you have higher free fatty acid levels. So there's a nice correlation between this in people. And if you look at the graph on the right, this is work from Elizabeth Parks, showing, using really elegant tracer methods, that the majority of fatty acids that are in the liver under conditions of fatty liver disease are derived from this circulating free fatty acids that come from adipose tissue. And indeed, if we looked at liver fat in our ALK7 loss of function and Inhibin E knockout mice, we see an increase in hepatic lipid accumulation in all of these models. And so these are germline knockout mice, right? There could be compensation that happens. So we were quite happy to see this recent paper that came out in PNAS where they used a pharmacological approach. So what happens if you treat therapeutically once animals are already in adulthood or maybe already under conditions of fatty liver with a neutralizing antibody against the ALK7 receptor? And they found almost exactly the same phenotype that we see in the knockout animals. So reduction in epididymal white fat, maybe a slightly reduced reduction in subcutaneous fat, just like we saw, yet an increase in hepatic lipid accumulation, an increase in insulin and impaired insulin tolerance. So just in the last two summary slides, I want to show you what I think is going on, what our working model is, and why we might have this disconnect between what we see in mice and what we see in humans. So we think that this hepatic inhibin E is actually acting as a lipid sensor in the liver and a feedback regulator of adipocyte lipolysis, such that inhibin E is elevated in the context of hepatic lipid or free fatty acid exposure. And then it sends a signal to the adipose tissue to say, hey, we've got enough fat going on here in the liver. We need to suppress lipolysis in the adipose tissue. And so you get this sort of homeostatic loop. And if you disrupt this axis, either by removing the inhibin E ligand or blocking the ALK7 receptor, that leads to now a lack of that signal coming to adipose tissue to shut down lipolysis. You get an increase in lipolysis, spillover of fatty acids to the liver, and increased hepatic lipid accumulation. And so the things that are consistent here between the humans and mice are the reduction in visceral adiposity. So this improved adipose distribution phenotype is conserved across the species, as well as the increase in adipose tissue lipolysis. And what's different is, as I mentioned, in humans, we get this reduction with type 2 diabetes risk, whereas in the mice, we actually see an increase in impaired insulin sensitivity and an increase in fat accumulation in the liver. So what accounts for this? I think one leading hypothesis is that it's a matter of time. So again, there's been another paper, there's a lot of papers that have just come out in the past couple of months. It's quite an exciting time for this mechanism. There was just another paper out in JCI Insight, which also treated with a neutralizing ALK7 antibody for a long period of time. And though they did see this transient increase in hepatic lipid like we see here, if you continue dosing over time, it's mitigated. So it could be that this is, you know, a response that happens acutely and maybe will dissipate over time. Another possibility is that there are species or strain differences in the amount of ALK7 in different adipose tissues between the two species. And maybe humans can handle the excess ectopic lipid hitting places like the skeletal muscle by increasing fatty acid oxidation, for example, maybe better than a mouse might be able to do. So with that, I will just wrap up and thank John, is a great Pfizer postdoc who's just wrapping up, and this work was just published. It's available online 2 weeks ago in Molecular Metabolism if you want to dig into a lot of the experimental details. There were many members of our IMRU team that worked on this as well, especially Trent, Barb, and Joanne, who were instrumental to analysis of all the different knockout lines that we had. And a special thanks to Mike Check at UMass, who was part of John's thesis committee. And I've put a link here if there are any trainees in the audience that are interested in the Pfizer postdoc program, I'm happy to tell you more about that at the break. Thank you.
Thank you, Dr. Bence. Do we have any quick? Hello?
This is very interesting. So you talked about lipolysis in the adipocyte. So what other targets genes does ALK7 have? Is there like a transcriptional signature for ALK7 activation in adipocytes that involves things outside of lipolysis?
Yeah, that's a great question. So of course I mentioned the SMAD phosphorylation. So in theory, you might get more fibrotic adipose tissue if you activate the ALK7 pathway. So again, potential benefit if you have a neutralizing antibody to prevent that. There are also changes in ATGL, hormone-sensitive lipase, so there could be a direct transcriptional downregulation of lipases when you activate this receptor. And the other things we see are things, beta-3 adrenergic receptor, for example, is also seems to be downstream of this receptor.
Kendra, that was a really elegant work. I really appreciated it, and somewhat familiar with the work myself. But it's very clear what the animals are telling us, and it's also very clear what the humans are telling us as well. So, you know, do you feel as though the interpretations that you get from the animals can really, you know, warrant moving a project forward, or not? Like when the human genetics is kind of more powerful, I would say.
Yeah, no, I mean, this is what we struggle with every day, right? It's hard when you generate data like this that would suggest maybe a no-go, right? To move this forward. But I think that I would be in favor of continuing to investigate here, and it might be that we get the answer in people with the antibodies that are out there. And you know, like I said, there could be transient acute increases in hepatic lipid accumulation, but if they're transient and they're not pathogenic, maybe this is something you just need to dose through and get over the hump to achieve that ultimate benefit of metabolic protection. So it's a tough. Yes, yeah.
We have our last question up here.
I can hear you.
Fantastic thought. Thank you. I have a question. My group studied the adipocyte genesis, and yes, those circulating free fatty acid is cause NASH, but also why for the brown fat genesis. So I was wondering if the knockout model has any effect or changes in the cold sensitivity for brown fat adaptive like adipocyte genesis.
Yeah, that's a great question. There's actually a Cell Reports paper that reports a role for this axis in brown fat. We conducted, I should have mentioned this, but we did conduct a lot of our animal studies at thermoneutrality to try and limit the contribution of browning and brown fat thermogenesis to the phenotypes we showed here. So I don't think it's a big contributor here, but certainly in rodents, I think there could be a role also in brown fat.
That's real. It's not kind of mimicking
yeah.
the environment people got.
Yep.
Thank you.
Mm-hmm. Thank you.
All right, let's thank Dr. Bence one more time. And now we'll have our micro talks. Great way to get the trainees involved. They are the only thing between us and lunch, so we will do a really good job of keeping them to 3 minutes.
Our first microtalk speaker will be Dr. Mark Sherman, who's a gastroenterologist and instructor at the Mass General Hospital and a postdoctoral researcher at the Goessling lab at Harvard Medical School. Please give it up for Dr. Sherman.
Hello, can you hear me? Okay, perfect. The accumulation of unfolded proteins in the endoplasmic reticulum elicits a stereotyped response, of which one example is XBP1. And this stereotype response then drives production and expression of molecular chaperones like HSPA5 to help address the burden of unfolded proteins in the ER. This is collectively called the unfolded protein response. And there is a suggestion that this is linked to NASH in the following way. If you knock out both XBP1 or HSPA5, the signaling molecule or the chaperone respectively, that's sufficient to induce fatty liver and sensitize to NASH when coupled with high-fat diet. In our lab, what we've been doing is giving wild-type mouse high-fat diet for very prolonged periods of time, to which they develop simple steatosis, NASH, and even HCC at very long time points. And when we examine the livers by single nuclei sequencing in the hepatocytes, we find that the unfolded protein response is actually markedly depleted. In light of the knockout data, this suggests that the unfolded protein response loss is maladaptive and potentially sufficient to explain induction of NASH in wild-type mice. And what gets even more interesting is when you look at the protein level expression. I'm showing you here high-fat diet mice at 6 months. In white is XBP1, that signaling molecule as a part of the unfolded protein response. It is massively depleted in zone 3, which is the bottom right corner here, which is the same region in which fat accumulates in the liver in NASH. And so if we sort of computationally exclude cytoplasmic staining and just show you nuclear localized XPP1 where this transcript factor is supposed to work. Sorry, the laser is not working. But anyway, you can see there's a marked loss of nuclear localized XPP1 in the top figure. And then in the bottom, we see the same pattern for HSPA5, which is the chaperone molecule, again, is lost in the right and bottom plot, is lost where fat accumulates in the liver. And these are quantified here at the right zonally. So I think what I shared with you today is that XPP1 and HSPA5 are two key components of the unfolded protein response. They're sufficient to cause fatty liver. What we're finding is in wild-type mice, which have these factors intact, nevertheless sort of phenocopies loss of these molecules, especially in zone 3, which is where we think NASH originates. So with that, I will thank you for your time, the opportunity to speak, the symposium organizers, Wolfram, and your attention. Thank you so much.
Thank you, Mark. Next, we have Dr. Arijit Gattu.
Hello, everybody. My name is Arijit Gattu. I'm from Roncon Lab. I'm one of the endocrinologists at MGH. We are interested in understanding hepatic insulin resistance in a type 2 diabetes using iPS cell-derived hepatocytes. Briefly, about insulin signaling, we all know that insulin is central in mediating metabolic actions in the liver. Insulin binds to insulin receptor and activates a cascade of kinases that would have various biological actions, which would be gluconeogenesis, glycogen synthesis, and lipogenesis, and others. But the primary driver of insulin resistance remains unclear, partly because there are many intrinsic factors as well as extrinsic factors that affect the signaling in a hepatocyte. We have been interested in the intrinsic factors shown here, both genetic and epigenetic. To answer that question, we have a hypothesis of understanding cell intrinsic alterations that would affect the metabolic pathways underlying the drivers of type 2 diabetes. To model that, we have used human iPS cell-derived hepatocytes from patients with type 2 diabetes and differentiated them into hepatocytes. What we found was very interesting. In the absence of any extrinsic factors, no fatty acids, no cytokines, type 2 diabetes, IHEPs, show selective insulin resistance, where insulin loses its ability to suppress gluconeogenesis and continues to exert its effect on lipogenesis. Shown here is a gene expression of PepCK1 and fatty acid synthase. On the right side is insulin-stimulated de novo lipogenesis. We next asked if we could do a phosphoproteomics to understand this better. Shown here is insulin-stimulated increased phosphorylation of a heat map of control and type 2 diabetes hepatocytes from basal insulin, both males and females, broken down into 3 clusters for the sake of convenience. The top cluster has equal increase in phosphorylation in both control and type 2 diabetes. An example of this is a TSC2. In the middle cluster has AKT2, where you have a loss of signal in type 2 diabetes. And the bottom cluster, where you have an increase in phosphorylation in type 2 diabetes. An example of that is BOP1, one of the serine sites. So now we wanted to understand what are the upstream kinases that drive insulin resistance. In collaboration with Luke Antley's lab at Dana-Farber, who recently developed a method to annotate all the kinases to substrates. What we did here is define the kinome of insulin action. Shown here is a volcano plot of all the kinases, and some of them are familiar to this audience. For example, the P70, P90, AKT1, PDK1. Using this as a background, we defined the kinases that are perturbed in type 2 diabetes. Shown here are some of the kinases that are not or have no activity in type 2 diabetes compared to controls, such as AKT2, as we all know, and some of the novel kinases. And on the other hand, where you have an increased activation in type 2 diabetes compared to controls. In summary, type 2 diabetes, IHEPs, revealed disruptions in insulin signaling pathways related to AKT2, CHEK2, ROC1 and 2, and MST4 kinases. And thanks for your time, and thanks to all the organizers.
Our next speaker is Iris Su. She's assistant professor for radiology at the Mass General Hospital and faculty member at the Martinos Center for Biomedical Imaging.
Sorry, need a little bit technical support. So I can get started first. Thank you for the introduction. So Yeah, perfect. So our lab has been interested in imaging fibrosis because it's a major determinant of morbidity and mortality in most chronic liver diseases. And there's a lack of non-invasive methods allow us to detect end-stage fibrosis with high sensitivity and specificity. So a hallmark of liver fibrosis is actually the excessive amount of extracellular scar tissues, particularly type 1 collagen. So this drives us to develop the molecular MRI probes that bind to type 1 collagen, for example, the EP3533 or CM101 here. So we have been validating this molecular MRI technique in various mirror models of liver fibrosis and injuries. So here we can see the collagen MRI allow us to detect end-stage fibrosis with a linear correlation to the biochemically determined collagen amount using the hydroxyproline analysis and also with the Eyshang score of fibrosis. Additionally, it allows us to detect treatment response, and for this case, this is looking at an intrahepatic response, intrahepatic heterogeneity in the rat treated with rapamycin, and we can see imaging allows us to see this, partial response to the treatment. And before the collagen cross-link and deposited, the upregulation of the lysoxidases triggers the formation of the extracellular aldehydes, particularly lysin. So this also triggers us to develop this a series of molecular MRI probes that is targeted to the lysin. And here in this model, we can in this study we have shown that using the lysin probe and for molecular MRI, we can allow us to detect early detection of disease as early as 2 weeks animals on diet, and also for early readout of the treatment response, like we showing here. And we can also see here the lysin MRIs actually correlate nicely with the with the fibrogenesis markers, not with the fibrosis markers. This shows the specificity of the analyzing MRI to the disease activity. And we didn't stop here because MRI is such a versatile imaging modality with a multi-parametric readout capability. So we can combine into this different MRI readout, including the collagen MRI, analyzing MRI to read out disease activity, and also with elastography, with fat fraction quantification and the T1 measurement, all in this one MRI imaging session in animal models. And here we can see we can detect both the disease progression and also treatment response. So more details about this one, I have a poster outside as a poster number 16. And we also didn't stop here, not only using the one imaging modality, and we can combine multiple modalities together. So this is an example of the integrin, which is also increasingly expressed in by the activated hepatic stellar cells. And we can using the integrin targeted PET imaging to assess the drug targeted engagement. And also in this case, we combine both PET, integrin PET, and also with collagen MRI to assess the targeting expression, also to using the collagen MRI to have a treatment readout, which correlate nicely with the histology. And with that, I thank you very much. I want to highlighting the multi-parametric, multimodal imaging methods that allow us to non-invasively and comprehensively characterize the disease, and also as a really sensitive tool for us to assess the treatment response. So thank you all.
Okay, and so with that, I'm going to introduce our next speaker, which is Dr. Markus Grompe, who's a professor at the Oregon Health and Science University. He's a world-renowned physician scientist who's clinically active as a biochemical geneticist. His research covers three main topics: cell and gene therapy for genetic liver diseases, Fanconi's anemia, and beta cell replacement for patients with diabetes. In the field, he's very well known for his establishment of the FNRG mouse, and has commercialized that mouse. And he's now using it to explore different replacement therapies using humanized livers. So his topic today is cell therapy with genetically enhanced hepatocytes. So let's give a warm welcome to Dr. Grompe.
Thank you. It's delightful to be here. Meet up with Win and Sangeeta and and Wolfram and all the organizers. Thanks to the ALF for a great dinner last night. Really appreciated that. And what I want to tell you about, I liked your joke, by the way, Harvey, about at the NIH we can only dream about disclosure slides, having something to disclose on the disclosure side. I think, that was among the many great jokes you had in your talk. So my dream is to help bring hepatocyte transplantation or liver-directed cell therapy to the clinic. We've used hematopoietic stem cell transplantation for many years successfully. Islet cell transplantation is coming along. And while orthotopic liver transplantation is a cure for many liver diseases, liver cell therapy is not yet to the point where we can apply it to patients. But I think we'll get there, and I'll tell you two parts to that puzzle today where I think the technological solution is to bringing this to the clinic. So for those who haven't reviewed hepatocyte transplantation recently, it's actually been done quite a bit with a really good safety record, over 100 human patients. The case reports you can find in the literature. There have been no properly controlled studies done yet to show efficacy, but in animal experiments, we can show that hepatocyte transplantation definitely can be beneficial in liver disease. It's been done in babies, newborns even, through the umbilical vein, and without major side effects. So we do know that it is safe to do this. And there are quite a few reports tantalizingly suggesting that biomarkers can improve after hepatocyte transplantation in humans and certainly in animals. So particularly in inborn errors of metabolism, which is my clinical field of interest. So the question is, so if this is so great and so has so much potential, so why is it not in the clinic yet? Well, there are three major obstacles to clinical hepatocyte transplantation. One is the cell source, and I'm not going to talk about that particular aspect today, but I do believe that there are ways to expand human hepatocytes that we could discuss if you want to, that are clinically actionable. But another big obstacle is that hepatocytes engraft at quite low levels. So when you try to put them into the liver itself, you're blocking the portal circulation and you cause portal hypertension. So you can only shoot so many hepatocytes into a liver before you start getting portal embolism and actually necrotic tissue damage. So we are definitely stuck at the less than 1% primary engraftment level. And then, of course, the source to date has been allogeneic cadaveric hepatocytes, which means you have to use immunosuppression. So what I want to do today is talk about 2 and 3. So problem number 2, low engraftment levels. Those of you who are familiar with the FRG or the FAH knockout mouse will come to appreciate that I believe that selection is the solution to low efficiency engraftment in gene therapy and in cell therapy. So we've been working on a way to select hepatocytes post-transplantation that could be clinically actionable. So the idea is you put in the cells, and then you use, like in the hematopoietic field, you use drugs to give the donor, the graft, a selective advantage, driving replacement to the level that you want it to. So we've developed a system that I believe is universally applicable. It should work in any liver independent of the genetic disease, should be safe enough for clinical use. Again, I always envious of our hematopoietic colleagues who worked this out in the 1950s and 60s, how to do this. So in order to make this happen in the liver, we need controlled liver damage. So I won't tell you about all the failed ideas, there were many, but we always wanted a hepatotoxin that could be used clinically, and people would say, okay, we'll let you do this. Ideally, it should be FDA approved. The mechanism of hepatotoxicity should be understood in great detail. You'd like it to be a compound that's been used a lot, including in kids. Ideally, you'd want a toxin that you could inactivate if things got out of hand. And ideally, it should also be cheap. Well, it turns out there is such a drug. I'll tell you about it in a second. But the principle of our interventions, of all of the things we've tried, is that you would have a drug that only, it's a protoxin. So it's not toxic itself. Something has to happen in the hepatocyte to make it toxic. This is usually a metabolic conversion. And then the general principle is, and this you could apply this to many different drugs, is that you would knock out or inactivate the enzyme that converts the protoxin to the toxin. And then that cell would be resistant. And you also need a toxic metabolite that acts cell autonomously, so doesn't have a bystander effect and goes into healthy cells next door. Okay, so it turns out this is the drug. And every little box of Tylenol has a liver toxicity warning on it. And I should just, we'll get to this. I always get the same questions about safety. Could you do this? Well, there are numerous clinical reports where people who just take Tylenol for fever or whatever, they measured their transaminases, and they're actually running around with elevated liver function tests and they don't even know it. So that's the kind of toxicity we're talking about, low-grade APAP toxicity that does not cause clinical liver disease. Okay, so how does acetaminophen become toxic? So its normal metabolism is through glucuronidation or sulfation, making it basically a stable kidney excretable metabolite. But a percentage of the drug gets converted to NAPQI, which is a highly electrophilic compound and can kill hepatocytes. The acetaminophen is converted to NAPQI by any one of 3 cytochrome P450 enzymes, 1A2, 2E1, 3A4. They're all zone 3 CYPs. Now, we knew that we couldn't really efficiently knock out all 3 CYPs. So we decided to target the cofactor, the obligatory cofactor for CYPs, which is NAD cytochrome P450 reductase. And the gene symbol is CyPOR. Okay, so the simple idea is we knock out CyPOR. Those hepatocytes can't make APAP into NAPQI, and therefore we should be able to select them with Tylenol. So here you have it. It's the same schematic, except now specifically talking about cytochrome P450, protoxin to toxin metabolism. And to test this idea, a graduate student in the lab and Vonada made a plasmid with CRISPR-Cas9 and then a POL3-driven guide RNA for CyPOR. We did hydrodynamic injection into wild-type mice. These are not FAH deficient. Let the hydrodynamic injury settle down, and then we put the animals, we treated the animals with acetaminophen, and then asked the question, what happens? So this is not transplantation, this is just knocking out, creating a population of CyPOR-resistant hepatocytes in that liver. So in the early days, we used sort of maximal APAP toxicity. We did intraperitoneal injection of Tylenol at the close to lethal dose. And you can see the initial transaminase responses. You get animals that go up to 10,000 within 6 hours after injection. And then we kept doing this twice a week. And by 10 weeks, there's no longer a transaminase response to the same dose of Tylenol. When we look at the percentage of liver cells that are CyPOR deficient by doing a molecular analysis, it's around 20%. There's about 60% of the DNA in the mouse liver is from hepatocytes, so do the correction. So about 30% of the hepatocytes are CyPOR resistant, CyPOR mutant. And if you do a CyPOR immunohistochemistry in these animals with and without Tylenol, you can see that the CyPOR tends to be enriched in zone 3. It's actually in every hepatocyte, but this stain doesn't show the zone 1 and zone 2 very well. But after Tylenol selection, you get this Swiss cheese appearance of CyPOR null clones that now make up 30% of the liver. So basically, this works. You can select CyPOR negative hepatocytes with Tylenol. And so we decided to do this in the context of gene therapy. This is published. But more recently, we've done it in the context of hepatocyte transplantation. So what we do is we take wild-type hepatocytes. We actually use a marker strain, MTMG, fluorescent red animals. We have a highly efficient in vitro procedure where we expose the cells to CRISPR-Cas9 in vitro. This is a very gentle procedure that leaves the cells transplantable. And we wash them and we put them into a mouse. And in this case, we used a mouse model of a common metabolic disease, phenylketonuria. It's a PKU knockout mouse, which has a deficiency in phenylalanine hydroxylase. And this is the result, just came out in Hepatology. You can look up the details there. We put these cells into PKU mice. Then you can tell from the red curve that unless you do selection, this low dose or this less than 1% engraftment does not change blood phenylalanine levels. But we put the animals on an APAP diet, and after just 2 months, they are completely corrected. Phenylalanine is normal. And then we stopped the APAP at that point to ask the question, is this a stable cell, this CyPOR knockout cell? So we've gone out to up to 1 year later, and the Phe levels stay normal. So the CyPOR mutant hepatocytes do not disappear. They're completely stable over time. This is what the histology looked like. The donor cells I already mentioned are red from an MTMG mouse, so you can see them nicely. They're PAH proficient. And then, of course, the selected cells are CyPOR negative, so the red and the no CyPOR completely overlap. In this particular animal, 16% of the hepatocytes were from the donor that was sufficient to correct the blood Phe levels. We've done a titration, and we know that 8% enzyme proficient hepatocytes is enough to cure the disease in mice. So what about the side effects of this? There's quite a bit of data in this recently published hepatology paper. I showed you the initial experiments where we had massive transaminase responses, and people say, well, you can't do that. But what you see here in red are the transaminases in animals that have been put on the diet that was used in the PKU treatment paper. And we just mill in APAP into the chow. The mice eat it, and they don't really have liver disease. They just have a mild transaminitis. These are the same animals that are selected in 2 months. So they have no hyperbilirubinemia, no other markers of liver disease, and there's no mortality or even visible toxicity. The other question that comes up, well, if you inactivate CYP metabolism, they become unable to metabolize drugs. And so our answer to that is you don't need to replace every CYP proficient hepatocyte to get therapeutic benefit. So here in this PKU mouse that had 8% overall replacement, you can see the donor cells in zone 3. That's where you have apoptoticity. That's where the donor cells sit. And then co-staining with CYP2E1, the enzyme that's actually metabolizes APAP. And you can see most of the cells are still green. So they are CYP metabolism proficient, but we have enough healthy donor cells that are CYP negative to be able to have the therapeutic benefit. So if the disease you're treating requires 10% or less cell replacement, you can do this without sacrificing CYP metabolism. And this is borne out by a caffeine kinetics. Caffeine is metabolized by CYP2E1. The animals that were cured are in blue, and the ones that were didn't have APAP selection in red. And you can see that the area under the curve in terms of caffeine elimination by CYP2E1 is not different between the two groups. Okay, problem number 3, I have 5 minutes left, which is allorejection. So in this particular circumstance, we just borrowed everything from the T-cell people and the iPSC people. It turns out allorejection is fairly well understood, given the importance of it in transplantation medicine. That's not that surprising. Armies of immunologists have studied this. It turns out in a T-cell, if you knock out class 1 and class 2 MHC, and also put in a decoy for NK cells so that they don't attack the missing self MHC negative cells, that you can do transplant allogeneic cells and the immune system won't see them. So the immune system the T cells need class 1 and class 2. And then when those are gone, the NK cells can recognize this and normally attack these cells. But you can fool the NK cells by either expressing CD47, which is what Sana Therapeutics does. Others express non-variant HLA, such as HLA-E or G, and then the NK cell is happy and won't attack that cell. So we started this work when I was with Ambus Therapeutics, and recently I brought this to my academic lab. And the idea is simple. We'll engineer the T cells, not the T cells, the hepatocytes, the way people engineer T cells ex vivo using this procedure I already mentioned with CyPOR, and then ask the question, can you do allotransplantation? So here's the experiment. We use that MTMG wild type animal, and then we're going to transplant in the controls C57 into C57, a congenic transplant that should work. And then we will do a mismatch strain. So it turns out in my lab, we have the FAH mutation on both backgrounds. And we actually did this in the tyrosinemia animal model for the ease of detection of the transplanted donor cells. And we did controls where there was a mock RNP. We did HLA class 1, MHC class 1 only with beta-2 microglobulin knockout. And then we did the T cell maximum where we knock out beta-2 microglobulin, C2TA, which gets rid of class 2. And then we overexpress CD47 by like 100-fold using a lentiviral vector. The experiment, the result is very interesting and gratifying. So in the congenic, of course, you see these donor cell clusters. That's the positive control, showing that we can actually transplant hepatocytes. In the non-treated cells, there's no donor cell engraftment in the allogeneic recipient. Very surprisingly, beta-2 microglobulin alone worked. And it worked as well as the triple manipulation. So I didn't have time to show this. We've now done this multiple directions. This was initially a 2-month readout. We're now at 6 months out with stable engraftment of allogeneic cells. And of course, in the FAH knockout mouse, because you have selection, we can actually cure tyrosinemia with an allogeneic hepatocyte transplant using this approach. Of course, that's cheating because we're using selection. But in terms of the immunology, it's not cheating. So beta-2 microglobulin alone is sufficient to prevent allorejection in hepatocyte transplantation in mice. Now, there's a lot of sad stories about how mouse immunology did not transplant into human immunology. So I'm not willing to say that this is translatable without non-human primate experiments, but I know there are no experiments on the books showing that this missing self and K cell mechanism works in solid tissues. All of the published work is on blood cell transplantation. So it's quite possible that the liver microenvironment is not conducive to NK cells recognizing missing self. So here's my overall summary. I think hepatocyte transplantation will see the light of day. I think all of the 3 major obstacles, cell abundance, selection, or poor efficiency engraftment, and allo rejection are all solvable. And there are actually some companies who seem to believe the same. The Satellite Bio that Sangeeta is involved with. There's a company Ligenesis out of Pittsburgh, and there's a Minnesota-based company, Cytotherics, all which are interested in human liver cell transplantation. What I'd like to finish up with is highlighting the fact that hepatocytes are one drug for many, many genetic liver diseases. So it should work for PKU, for hypercholesterolemia, for all of those conditions. When I compare that to gene therapy and you think about the path, the regulatory path at the FDA, there you need a specific approval process for every single drug you make, which really slows things down and makes super rare liver diseases very unattractive commercially. This should work for even the super rare liver diseases for which there's only like, let's say 10 patients in the United States. And so I think this one drug for many diseases advantage, I think, is really the main advantage of hepatocyte transplantation over gene therapy. And with that, I'd like to acknowledge particularly Anne, who's about to graduate in early December, and Amita Tia-Bunche, who was a postdoc in the lab and is now an assistant professor. Thank you.
All right, I already see a question, yeah?
Markus, really fascinating data and beautiful work, and congratulations of having the courage to use an old toxic drug for this. I have a zonation question. So you showed, if I saw this correctly, that most of your CyPOR negative cells in the end are pericentral. And then if we think about the periportal pericentral gradient and differential expression of genes, do you think you can only fix genetic defects or enzyme defects that are expressed in the pericentral region? Or have you seen or followed your animals long enough that they essentially then populate, retrograde, and go towards periportal region where they might express different genes?
If you wait long enough, they start migrating. But that being said, of the diseases we're interested in, they tend to be expressed everywhere. Every hepatocyte has glycogen storage enzymes. Every hepatocyte has phenylalanine metabolism. So there might be, I don't know of a zone 1 specific genetic liver disease. I do know of zone 3 specific liver diseases. And for those, this will be particularly interesting. There's a genetic blindness called gyrate atrophy, which is due to deficiency of ornithine aminotransferase in just the pericentral area. And that's actually on our high to-do list because we actually have the ability to fix just those cells. So I know of zone 3 only enzymes. Most of the diseases we're interested in are in every zone of the enzyme.
At which time point after you induce injury with acetaminophen, would let me rephrase, would the outcome be different if you choose a longer time period between injury and testing the outcome? So how much time is there to conduct the hepatocyte transplantation?
So hepatocyte engraftment, moving from the sinusoidal circulation into the parenchyme takes several days. So we err on the side of being conservative and wait like 2 weeks. So we know that whatever cells are engrafted are stably engrafted, have started expressing their proper genes and so forth. So I think if you did it very soon after transplant, I think the cells would not be, they wouldn't be selectable because I think they're really not part of the liver yet. So I think you need to wait long enough until they're really seamlessly integrated into the parenchyme. But I don't have data. That's just the way we've been doing it.
And may I ask as a follow-up question, how would this be applicable to patients that have acetaminophen overdose and go into acute liver failure?
So I think this does not apply in an acute setting. This is really only for genetic liver disease. This is the approach we're taking. Yeah.
Okay, thank you.
Markus, can you clarify something that's always been confusing to me and seems to be some differences of opinion in this field? The question is, how do the transplanted cells, which I presume you're injecting into the spleen,
that's right, yeah.
How do they actually gain access to the hepatic globule? How do they get through the endothelial barrier? Are they going through fenestra? Are they going through tight junctions? And once they enter into the lobule, how do they actually connect to the existing anatomic structures, say, of the bile, canaliculus, and all that? There was a point several years ago where people had suggested that they were actually fusing with the endogenous host cells. In other words, each transplanted cell was contributing half a canaliculus to the other cell. Could you clarify this cell biologic conundrum?
So it's a miracle. It's like it reminds me of one of those old cartoons, and then it says, then a miracle occurs. So this is totally understudied. We do know that it happens. We've looked at specifically the fusion question. We've ruled that out using genetic markers. So I can tell you it's no, for example, we transplanted Cre-expressing hepatocytes into a reporter mouse, and we don't see activation. The Cre doesn't get into the host cells. So I'm clear on that. But other than that, why it is and how the hepatocytes actually migrate. So this could be very fruitful for young people. Only of the cells we put it into the portal circulation, at most 10% arrive where we want them to, right? So if someone could figure out a way to make that number go up by better understanding the initial engraftment events, I think that would be a big boost to the field. I think it's very important. We and others have not really gone there. Can't do everything, but for us until now, it's been good enough to just know it happens. But I think it would be very interesting to study that.
Wonderful. Let's thank Dr. Grompe one more time. And I'll invite up my co-chair, Jason, to introduce our next speaker.
Our next speaker is Dr. Gouon-Evans. And she is an associate professor of medicine at Boston University, and she is also the director of the program of Boston University liver biologist. For the past decades, she has focused on understanding the liver development and establishing therapeutic strategies for liver diseases using iPS-derived cells, derived models, or the mouse models, as well as the using lipid nanoparticles to deliver mRNAs for regenerative factors to treat liver diseases. Without further ado, let's welcome Dr. Valerie for her talk.
Thank you for the introduction. I would like to thank the organizer, Sangeeta and Wolfram, for inviting me and to give me the opportunity to share with you our work on the use of nucleoside-modified mRNA to alleviate the liver disease. So as you know, the liver is known for its fascinating ability to regenerate. But during, however, during a severe acute or chronic liver injury, this ability is compromised. So my lab is developing some strategies. Oh, right, doesn't work anymore.
The digital pointer went out.
Oh, really? Oh, okay. All right. So yeah. Oops. All right. So now, oh man. So my lab is developing strategy to harness liver regeneration by promoting the natural intrinsic liver repair mechanism. And those include hepatocyte-driven liver repair, in which hepatocyte proliferate to replace loss of the liver mass. And also the cholangiocyte-driven liver repair when hepatocyte proliferation is exhausted. So in this case, some cholangiocytes are believed to be liver progenitor cells, and as such, they proliferate and they can differentiate into hepatocyte. Cell therapy using primary human hepatocyte or patient-specific induced pluripotent stem cell-derived hepatocyte is also a strategy that we are investigating. So in order to promote this repair mechanism, we propose to use some key mitogen, hepatocyte mitogen. So for example, AGF and EGF, but also growth hormone and VEGF-A. So to do so, we decided to use a tool that allows us to express robustly, but stringently this factor to prevent any deleterious effect. So we decided to use a nucleoside-modified mRNA. And so those are in vitro transcribed mRNA. And they harbor modifications that have been pioneered by the Nobel Prize winner of this year, Katalin Karikó and Drew Weissman. So this modification prevent immunogenicity and increase mRNA stability and translation into protein. So these mRNA have many advantages. They are clinically safe, as they do not integrate into the genome, and they have been successfully used to generate the COVID-19 vaccine recently. So when they are complex with lipid nanoparticle, conveniently, they target the liver. And they allow control expression of protein with time, with a maximum of a week of expression. So here to validate this tool for the liver disease, we first injected mice with luciferase mRNA LNPs to confirm the liver specificity of the activity. And we also found that this activity lasted only 3 days, which is good. So we also identify the cells in the liver that are transfected, and we injected mice with EGFP, mRNA, LNPs for that. What we found is that virtually all hepatocytes are transfected, 70% of endothelial cells and 40% of Kupffer cells, while cholangiocytes were not transfected. So as a first therapeutic intervention, we demonstrated that the two key hepatocyte mitogen, AGF and EGF mRNA LNP, can rescue a chronic liver injury mouse model. So these mice were fed choline deficient thionine supplemented diet to induce steatohepatitis and hepatocyte death. And they were treated with 2 doses of AGF and AGF mRNA LNP during the recovery period. So we can see on the left that the mitogen actually rapidly restores serum ALT level just 2 days after the first injection. And here you can see that the mitogen treatment also eliminated steatosis that is still seen in the control treated mice. And consistent with this data, we found that in these mice, the serum level of cholesterol was inversely correlated to the degree of steatosis in the liver. So this data showed the clinical benefit of AGF and EGF mRNA LNP to eliminate cell death and steatosis, which are 2 features of NAFLD. So next, I would like to tell you a project from Anna Smith, a student in the lab. This project aims to harness cell therapy for liver disease. So here she transplants either primary human hepatocyte or iPSC-derived hepatocyte-like cells. And this is to treat the liver disease associated with alpha-1 antitrypsin deficiency, AATD. And for that, she used this mice NHG-PIZ that recapitulates the liver disease found in human. So in these patients, there is an accumulation of the mutated form ZAAT in hepatocytes that causes the liver disease. So here, Anna proposed that, and this mouse model recapitulates exactly that. So she proposed that stimulating the key regenerative pathway AGF and EGF in transplanted hepatocyte will improve survival, proliferation, and engraftment of human hepatocyte in an in vivo model that mimics AATD liver disease, which is NSG-PiZ. So in addition to this background, she wanted also to mimic the hepatocyte senescence that is seen also in this patient. So for that, she injected AAV P21 that expressed P21 in hepatocytes specifically. She transplanted 1 million cells and stimulated survival and proliferation of the donor cells by injecting AGF and EGF mRNA LNP twice a week for 2 weeks. So she collected the liver of 4 groups of mice, the control treated group, P21 treated mice, AGF, EGF only, and the combined treatment. She identifies the transplanted cells by doing immunostaining for human albumin, the human nuclei Q80 staining, EDU for proliferation. So what she found is that when, in the presence of P21, AGF and EGF, this condition is in blue. You can see that AGF and EGF improve survival, proliferation, and engraftment of the transplanted cells. And she did that by calculating the number of primary human hepatocyte cluster, the number of cells per cluster, or the number of proliferating cells, and also the percentage of human albumin area in the liver. So, and now she did a treatment for 5 weeks. And she found that AGF and EGF mRNA LNP leads to sustained engraftment of these cells. So here you can see the study increase of human serum albumin in these treated mice, and with a 37-fold increase in the treated mice compared to the control. And by that time, about 30% of the liver was repopulated with human cells, as you can see here with a Q80 staining. So, and importantly, this level of repopulation allow also to reduce the AATD liver disease burden. So for example, on the top, you can see a decrease of the polymers Z-AAT in hepatocyte, and this was quantified in the treated mice. Also a decrease of circulating serum Z-AAT, as well as a decrease of the liver damage with lower level of ALT. So in conclusion of this work, Anna showed that AGF and EGF mRNA LNP can improve cell engraftment and also mitigate the liver disease. So now she's applying this strategy to also improve human iPSC-derived hepatocyte to treat this disease. So lastly, I would like to tell you about this project from a postdoc in the lab, Fatima Rizvi. So she's using VEGF-A mRNA LNP to promote cholangiocyte to hepatocyte conversion in vivo and reverse also the chronic liver disease. So the rationale for using VEGF-A is that VEGF-A is a ligand for the receptor VEGF receptor 2, or called also KDR. And we showed previously that we identify actually an hepatic progenitor cells using in vitro pluripotent stem cell differentiation culture from human and macaque. And when we inhibited KDR in this culture, we actually decrease the ability of this progenitor to differentiate into hepatocyte. And we also identify this early progenitor expressing KDR in the mouse model, and we found that it can give rise to hepatocyte in adult. So this finding led us to speculate that the regenerating liver uses the same cue as a developing liver. So Fatima asks these two questions. Is VEGF receptor 2 or KDR expressed on cholangiocyte, which are a source of progenitor cells, following liver injury? And the second question is that then, can we use VEGF-A as a ligand to activate cholangiocyte to differentiate into hepatocyte and also rescue the liver function? So as expected, Fatima found that during liver injury, KDR is expressed on a subset of cholangiocyte, and she found that in chronic injury as well as acute injury, which was acetaminophen. So you can see the arrowhead that indicate these cholangiocytes that express KDR. So to address, to study her hypothesis, Fatima used the two different injuries, the acute acetaminophen overdose and the chronic injury I just told you about, the CDE diet. She injected all the mice with AAV P21 to induce hepatocyte senescence, because hepatocyte senescence is observed in all chronic liver injury in human, and also actually in after acetaminophen overdose. And she delivered VEGF-A using mRNA LNP. So she lineage traced the fate of cholangiocyte or cholangiocyte expressing KDR using this two mouse model with an inducible Cre expression in the tomato reporter mice. So upon tamoxifen injection, cholangiocyte become tomato, as well as hepatocytes that are generated from the cholangiocyte. So now what we are going to look at is, do we see tomato positive hepatocyte? So this is an acute injury, so acetaminophen model. So after acetaminophen injection, mice were administered 2 doses of VEGF-A mRNA LNPs. And here you can see on the left in control treated mice, so control RNA LNPs treated mice, very rare hepatocyte, tomato-positive hepatocyte, and they are indicated with an arrowhead. While in contrast, VEGF-A induced drastically the conversion of cholangiocyte into hepatocyte, as you can see here, all the hepatocyte, red hepatocyte, and the identity of this hepatocyte were confirmed with HNF4 alpha staining. And this was very, very robust. The VEGF-A induced 5-fold increase of this cell conversion compared to in the control treated mice. And it was also very consistent. The cell conversion was seen in all liver lobe of all the mice studied. So, and this is the data of the chronic disease, the CDE diet disease. So here you can see again, you know, not only VEGF-A could induce drastically the cell conversion, cholangiocyte into hepatocyte. You can see this large cluster of tomato hepatocyte here. That was a 5-fold increase compared to the control mice. And here, not only VEGF-A can do the cell conversion, but also completely revert steatosis that you can still see in green in the control-treated mice, but also reverted fibrosis. And this was still seen in the control in blue. And that was significant. So now she used the KDR lineage cell mapping, and she found exactly the same data after acute and chronic liver injury. So she could find this large cluster of tomato positive hepatocytes. The identity of this hepatocyte was confirmed with HNF4 alpha expression or CD26 expression, and that was significant. So now we wonder, you know, what is clinical relevance of this data? So what we were looking for is, can we find a cholangiocyte in human advanced liver disease that express KDR? So we collaborated with Alex Soto-Gutierrez, who provided us with specimen of advanced human cirrhotic specimen. And here you can see, so Fatima did some immunostaining, and in the fibrotic septa of this liver, you can see some cholangiocytes. So if you look at the frame number 4, it's here. So we can see some cholangiocyte KRT7 positive cells that also co-express KDR. And there were many of them, actually. And if we look at frame 3, we can see also these very interesting cells that are budding from this string of KDR positive cholangiocyte. These cells are also KDR positive, and they are very reminiscent to the hepatocytes that have been described by Van Les et al. At least a decade ago. So, and we could even find actually some cluster of what we think are hepatocytes that express KDR, and they were just at the margin inside the regenerative nodule. So to confirm KDR expression in hepatocyte, we also looked at KDR expression at the transcript level and protein level. So this time, Alex gave us some frozen pellet of hepatocyte purification preparation of 9 different advanced cirrhotic specimens and 3 normal specimens. So we confirmed that, I mean, he has published this technology, but we still confirm that they were hepatocyte and they express similar level of HNF4 alpha transcript. Interestingly, we also confirmed that the level of the mature form of HNF4 alpha was down-regulated in this patient. But importantly, looking at KDR transcript actually were upregulated in the patient sample, and it was also, the protein was also present. So that was very exciting. I mean, it suggests maybe that this hepatocyte derived from the cholangiocytes that express KDR in this human specimen. And what it means also is that VEGF-A mRNA LNP may serve as a therapy to potentialize cholangiocyte-driven regeneration in this advanced liver disease patient. So I would like to finish here. So I hope I convince you that mRNA LNP is a great tool to harness liver regeneration. And I showed you that AGF and EGF mRNA LNP can induce hepatocyte-driven liver repair by promoting proliferation and also eliminating steatosis. Show you that VEGF-A mRNA LNP harness cholangiocyte-driven liver repair and also decrease the steatosis and fibrosis, and that AGF and EGF mRNA LNP can improve cell therapy for liver disease. So I would like to acknowledge that actually the whole lab work as a team to really establish the mRNA LNP tool for liver disease. And I would like to acknowledge Fatima Isley and Anna Smith, who did the work I presented specifically to them. Our collaborator, Drew Weissman, Norbert Pardy, and Hien Dang, who provided us with mRNA LNPs for these old studies, and also Dong Gunshin and Alex Soto Gutierrez, who were actively involved in Fatima's project, funding sources. And yeah, just wanted to say that I'm hiring right now a postdoc, so if you're interested, please come to me. And thank you very much for your attention.
Thank you. It's a great talk. Questions? Yeah, of course. Sure.
Fantastic talk. I have one question is for both segments. First, with the LNP VEGF, what is the isoform in LNPs of the VEGF? 165 or 121, 191? That's first. Second, what is the source of the VEGF in vivo? Is it cholangiocyte producing? They have clearly KDR, the receptor. Where this VEGF is coming from? Sinusoidal endothelial cells, other cell type? Yeah, that's on the VEGF-A, on the EGF and AGF. So when you give this AGF and EGF in vivo, they're gonna be also supporting the resident the host hepatocyte as well. So how they're gonna be preferentially helping the donor hepatocyte that engrafted to grow more competitively? So
right.
Maybe too much
Okay,
question.
let's see if I remember all the questions. So first
VEGF
one is
isoform source.
VEGF-A 165. Yes.
165?
Yes,
Okay.
165. So the cells in vivo that would provide VEGF-A, so there are studies that looked at that, and cholangiocytes themselves actually, and we found also, we did some single cell RNA seq analysis of this liver, and we found two different populations of cholangiocytes during the after injury. And some of them express KDR, so that was nice to find by transcript level also. And some of them actually express, the other one express VEGF-A. So that was very interesting.
They almost act as an endothelial cell.
Yeah.
In terms of KDR signaling.
Yeah. But there was this paper that just came out a few months ago, and they show that stellate cells after injury induce also VEGF-A expression. So they could be also the source for VEGF-A. For your third question, so here, because in our model, we are mimicking what is happening in the human disease, which is senescence. And senescence, actually, it's quite important. The percentage of hepatocytes that express, for example, one way to senesce, it's expressing P21. The percentage are quite high. And recently, there was this paper who published, different kind of liver disease, chronic liver disease, and the percentage of P21 expressing hepatocyte. So because of that, so it's something that happened in humans. So that's why we mimic that in our model. And because of that, when we provide AGF and EGF, you know, it will preferably activate the transplanted cells.
So the donor cells are better prepared to take help from the LNPs.
Yes, I mean, it was still a question we had, you know, when we started these experiments, and it seems to work this way.
Wonderful, Zoe. Thank you so much.
Thank you.
We have last question. Do you want? Okay, due to time, yeah, let's thank Valerie again. Okay. Before we jump into the micro talks, we actually have a special program that Miss Lauren Steele, yes, yeah. Thank you. The CEO from ALF, she is going to share her kind of like thoughts and.
I'm just finishing up. Normally I have a really big mouth, so you don't need this. But anyway, I'm just finishing up my 3rd year at the American Liver Foundation. And I tell you, what drives me every day is research. And it's so exciting to have you all here and to continue this amazing tradition, this Irwin Arias Symposium. You know, we're glad we're back in person, and we are so grateful for the leaders who have made this possible. Wolfram and Sangeeta, and where is the amazing Heather Fleming? Heather, we are so grateful to Heather's leadership, and thank you so much. And of course, we are so grateful to Dr. Wynn Arias, who had the vision over 40 years ago to partner and create the American Liver Foundation for the benefit of patients. And we're so grateful to Luba as well for her history with the American Liver Foundation. Also want to acknowledge in the room one of our national board members, Dr. Ratanya Carr, and grateful that she's here. And I just wanted to share a couple of things about the American Liver Foundation. We are stronger than we have been in most of our history. We are focused in a number of key areas. One is public health. You know, there's so much we must do in screening people for liver disease. We've got 80 to 100 million people in this country with liver disease, particularly steatotic liver disease, and we must do more. And we started a program a few years back, our Think Liver, Think Life initiative. We're in 22 states. We're going to be bringing on another 10 states. And by the time we hit our 50th anniversary in a few years, we're going to be in all 50 states, raising awareness, education, and screening people for liver disease. We're also leveraging that to public policy. I was actually in Washington, D.C. and at the federal Capitol just yesterday and the day before, talking to legislators in the Senate and the House, making sure they understand we need to do more in liver disease. And we had a really productive trip, and I'm excited to say I believe the government's going to do a lot more for this growing epidemic. We're also doing a lot in transplant. You know, there's a lot of people sitting on waiting lists for transplants, for livers, and how beautiful that this organ regenerates. And we need to do more as an organization in encouraging living donor liver transplant. And you're going to see so much more from the American Liver Foundation moving forward. And last but not least is research. We are so very excited that we resurrected our research program after a hiatus around COVID. We got a record number of applications this year. If any of you applied, thank you. Our review grants committee is going to be taking a look at everything, and hopefully we will be funding a significant amount of research this year and many more years to come. And I wanted to share, we have a new addition to our team, Dr. Ellen Jordan. And is Ellen in this room somewhere? There she is. Dr. Jordan joined us earlier this year. She's a trained immunologist, human biologist, and we're just thrilled to have her on board leading our efforts. So also want to thank our team for partnering so closely with the leadership here in Boston. And we just thank you all so much for all you're doing in liver for the benefit of patients. There's so much to do, and we all hands on deck, and we're just grateful that you have chosen this discipline to work and perform miracles for the benefit of patients. So thank you, and go liver!
Thank you. Thank you. Okay, we are going to have the micro talk. So in this session, we are going to have 4 speakers. The first one is Liliana Mancio. So please, the floor is all yours.
I have a mic. What do we need to do?
I'm here. I'm sorry.
No, that's
there
okay.
we go.
All right, we can hear and see you. I think you can go ahead now.
Okay. Thank you for this opportunity. I'm Liliana. I'm based at the Institut Pasteur in France, and previously I was a trainee in Sangeeta's Bhatia laboratory. In my short talk, I will be giving you a glimpse of our recent advances to understand host pathogen interactions, and our pathogen of interest is the malaria parasite Plasmodium vivax that you can see depicted with the green color here in this image. And as you might know, vivax infections, there's a mix of parasites, some that are large and actively replicating, and other parasites are very small, and they remain in this non-replicative state for several years. And are those tiny parasites that are responsible for malaria relapses. These dormant stages of the parasite, they were discovered in the 80s, and yet the only way for us to classify them is measure their size under the microscope. So to improve on that, we turned into single-cell transcriptomics, and shown here, this is the pipeline we use to employ to do dual sequencing of the parasite and the host cell. And I wanted to highlight the in vitro liver system that was developed in the Bhatia lab and enables the culturing of the parasites for several weeks. And also wanted to highlight the fact that we used a targeted approach that yielded very good coverage of the parasites. We obtained nearly 1,500 individual parasites that are clustered according to their developmental stage, for which now we have transcriptional signatures. For the sake of time, I'll be focusing on the dormancy signature. In this heat map, you can see the list of the 10 genes that we found to be upregulated in the dormant parasites, and that we use to define our dormancy score that essentially enabled us to identify these two clusters as being the dormant parasites. So next, we use this same scoring strategy to identify the hepatocytes hosting the dormant parasites. We found that they cluster in this tail, and you can see them in green. And the genes contributing to these differences. So now we'll be comparing the hepatocytes hosting a dormant versus a non-dormant parasite. And there is alterations in various functions related to the metabolism and the immune genes. They are suppressed and suggest that the cell is also in a dormant state. So currently in the lab, we are investigating this further to understand whether this is an active process induced by the parasite or from the host cell. And that's it. Thank you.
Wonderful. Thanks so much. And now we'll go on to our next speaker, Iris DeYoung. You want to go ahead and share your screen?
Yes.
All right.
Shared, I'll put presentation mode.
That looks great. Go ahead.
Great. I'm Arias DeYoung. I'm a postdoctoral researcher at the University of Pennsylvania in the Wells Lab. And we're studying pediatria. Pediatria is a cholangiopathy affecting the extrahepatic bile ducts of newborns, leaving the ducts completely atretic at an age of 1 to 3 months of life, necessitating surgery at that point. The cause is unknown, but the initial injury is thought to be prenatal. And we questioned, how does the extrahepatic bile duct respond to prenatal injury? We first looked at normal and adult bile ducts. And strikingly, we found this white layer of hyaluronic acid, HA, located directly around the lumen in fetal ducts. Whereas in the adult, there was only a small layer of hyaluronic acid located directly around the lumen, with collagen taking up the entire bile duct wall. Whereas in the fetus, there is only collagen at the outside of the HA layer. As a model of fetal injury, we used fetal sheep, and we compared fetal sheep that underwent normal gestation with fetal sheep that were taken out of the maternal womb and put in a biobag, which is an artificial womb that allows for modification of oxygen levels, and we applied hypoxic conditions. So looking at liver sections, we found intrahepatic bile plaques together with a significantly increased conjugated serum bilirubin level, and this indicated cholestasis, and that our model of prenatal ductal injury actually worked. Switching to the ducts themselves, we saw in the control samples a white layer of HA around the lumen, which we expected. And in the hypoxia group, we saw a significantly expanded HA layer. In parallel, we found an increased volume and incredible hyperplasia of the peribiliary glands, which are these glandular structures in the ductal wall of the hypoxia group. We wanted to focus on this increased and prolonged deposition of HA that we found in the hypoxia group, which is consistent with the fetal damage repair response, part of a fetal wound healing program. So going back to the disease, biliary, we found that in BA remnants, almost the entire atretic bile duct was composed of HA, comparable to our fetal hypoxic ducts, and indicating that HA plays a prominent role in the BA pathophysiology. These data, together with other in vitro and ex vivo data, and the notion that HA is capable to swell even up to 1,000 times its own volume, led us to the following working model. After prenatal injury, the HA layer swells, effectively compressing the collagen layer at the outside and narrowing the lumen, eventually leading to cholestasis and obstruction. In conclusion, the fetal extrahepatic bile duct is composed of this white layer of HA located directly around the lumen, which swells upon damage, being part of the fetal wound healing program, together with increased peribiliary gland volume, leading to a narrowing lumen and contributing to cholestasis and obstruction, potentially eventually leading to biliary atresia. Thank you.
Thank you. Our next speaker is Nidhi Jailong Sakrikar. Please share your screen.
Hello, everybody. Yeah, you can
No,
see
it's
my
correct.
slides. Yeah.
Yeah, right now it's
Thank you. Yeah, thank you for this opportunity. I'm Nidhi Jalan Sakrikar, working at Mayo Clinic, and I'm going to just share a snippet of the project where we are looking at neutrophils in the pathogenesis of primary sclerosing cholangitis, or PSC. PSC, we know, is an immune-mediated cholangiopathy, and studies have shown that there are immune cells shown here, like macrophages and T cells, located in the proximity of the bile ducts. Recent study has also shown that there has been substantial neutrophil infiltration observed in the PSC bile ducts compared to the non-PSC control. However, the role of the neutrophils in the PSC, as well as the mechanism of infiltration, are not known. When we utilize immunofluorescence in our PSC liver tissues, as well as a non-PSC liver tissues, what you can see here in red is the MPO, which marks the neutrophil, whereas in green are the bile ducts marked by CK19. And you can see the neutrophils are in the close proximity to the bile ducts. This was also recapitulated in all the mouse models that we utilize in our lab for studying PSC. This is a diet-induced DDC model, as well as a genetic MDR2 knockout mice. And again, we see these MPOs, positive neutrophils, in close proximity to the bile ducts. Neutrophils are known to induce ROS and cause oxidative stress in the tissue. So we utilized, again, immunohistochemistry for 8-OHDG, which is known to mark oxidative lesions. And again, compared to the chow, we can see an increase in the oxidative stress in the DDC and the MDR2 knockout mice. To understand the mechanism of the neutrophil infiltration, we isolated primary cholangiocytes from our mouse models, TDC and the MDR2, and compared them to the primary cholangiocytes from the chow. And we can see that we have this increase in the cytokine expression that are known to recruit immune cells. We utilized our in vitro model to understand the mechanism, and we focused on this CGAS sting pathway, which is a known instigator of inflammatory pathway. So we use LPS to instigate an inflammatory response, and you can see that we have the CXCL1 increase, which was inhibited when we use a sting activity inhibitor, and I'm only showing you CXCL1 and CCL2. We also utilized this conditioned media from these cholangiocytes, and we looked at neutrophil chemotaxis. And again, we can see that in presence of the sting inhibitor, we were able to reduce the chemotaxis of the neutrophil, suggesting that this increased pro-inflammatory cytokine expression in cholangiocytes is dependent on sting. So our current working model is that stress to cholangiocytes activates the CGAS sting pathway that can cause cytokine release, such as CXCL1, which is important for neutrophil recruitment. The presence of neutrophil results in the degranulation that results in the oxidative stress that can further perpetuate the inflammation and the fibrosis seen in the PSC. So we are currently testing the application of the pharmacology inhibiting sting in the mice models. With that, I'd like to acknowledge my group, as well as the other members of the lab and my mentors at the Mayo Clinic and the peers and the source of funding. Thank you.
Thank you so much. And now we'll conclude with our final micro talk with Sean C.
Okay, so good afternoon. I am Sean, a PhD student from UT Southwestern Medical Center in Dr. Hao's lab. So many mosaicism occurs in many regenerative tissues, including liver, and it is highly related to disease progressions, especially for cancer. So our goal is to understand how adaptive immune system regulates evolutions by asking which mutations are selected for, what clones are cancer consequences, and what type of cells give selected pressure. Okay. So next, to study this in vivo, we established a system called Mosaic. In the system, we use adeno-associated virus to deliver a pool of guide RNA into Cas9 mouse to generate so many mosaicism in the liver. And to trace down clonal fitness, we introduce sleeping transposome system which integrate gRNA into the mouse genome, and then allow us to read out gRNA abundance, which is correlate to the clonal fitness using next generation sequencing. We designed a gRNA library targeting roughly around 600 genes that is related to cancer development. To study how immune system influence clonal selections, we perform mosaic on immunocompetent versus immunocompromised mouse. At the same time, we have a second screen in which we compare between normal immunity to enhanced immunity by giving immune checkpoint inhibitors. From our screen results, we saw many genes showing different clonal fitness in the context of different immunity. Among them, PAFH1B2 knockout clones showed different clonal fitness it showed less fit when immunity is intact. And this negative correlation indicates that the PAFH1B2 knockout increased immune surveillance and also sensitized the clone to the checkpoint treatments. So to uncover the mechanism, we tested how PAFH1B2 influenced PD-L1 expressions, considering PAFH1B2's function as an acetyl hydrolase, plus previous studies suggest that the deacylations of PD-L1 permits nuclear translocations. We hypothesized that PAFH1B2 can deacylate PD-L1 that promotes nuclear transitions and also the PD-L1 immunosuppressive activities. Our results indeed show that when we delete PAFH1B2, it increased the acylations of PD-L1, promotes lysosome degradation, and also reduces nuclear translocations. So in summary, we established an in vivo CRISPR screen to study the impact of adaptive immune system on so many mosaicism in the mouse liver, and we identified novel genes regulating immune surveillance of so many clones. So thank you for your time, and we're happy to answer any questions from the email. Thank you.
So to introduce myself, my name is Constantine. I'll be the session chair along with Kay for the last session of the day. As a reminder for anyone attending on Zoom, you can feel free to take a look at the QR code for the session program that will imminently be appearing behind me. For people attending in person, there was a phone found in the bathroom as well as a bracelet. So if you're missing one of those, please check at the security desk. Without any further ado, I'd like to also introduce our first speaker, Dr. Rotanya McCants-Carr, who is the Cyrus E. Rubin Chair and Division Head of Gastroenterology at the University of Washington. As a clinical hepatologist, she specializes in the care of patients with fatty liver diseases. As a basic science researcher, her lab uses complementary in vivo and cellular approaches to investigate the mechanisms of insulin resistance in MASLD and alcohol-associated liver disease. In addition to her many accomplishments, she was honored in 2020 as one of Cell's most inspiring Black scientists.
Thanks so much, Constantine. That's me. I am between you and wine, I hear, so I'm gonna make sure I stay on time. Again, I'm Rotanya Carr. I'm so thrilled to have been invited this year to speak to all of you. So thank you to our organizers. Thank you to the ALF, and thank you to all of you for spending your day here. I'm going to be talking about lipid metabolic regulators of steatotic liver disease. And those are my disclosures. The bottom is the most important disclosure that you should remember. And so that I don't forget the most important part of all of my talks, I always start with my thank you slide. This collage are all the people who have worked with me through the years, my lab mates who really have done so much work in helping us advance liver disease research. So newsflash, many of you already know, fatty liver disease is no more. It is now steatotic liver disease, or SLD, and non-alcoholic fatty liver disease is also of the past. It's now, breathe, metabolic dysfunction associated steatotic liver disease. MASLD is how it's pronounced. And I will be using those terms through my talk. And I was so excited to be part of the nomenclature process because steatosis was elevated. I love steatosis, and now steatotic liver disease is how we talk about the majority of our metabolic liver diseases. In the circle are the diseases, at least experimentally, that my lab investigates. MASLD, alcohol-associated liver disease, and the combination condition, which is now MET-ALD, so a combination of metabolic liver disease and alcohol-associated liver disease. And it is my view, and those of my lab mates, and hopefully all of you by the end of today's session, that understanding lipid metabolism is key to understanding the pathogenesis of steatotic liver disease. And this understanding really helps us on the macro, the meso, and the micro levels. So for example, when we want to have answer questions clinically in terms of finding diagnostic tools or therapeutics, lipid metabolism helps us. When we're talking about physiology, which is a major investigation within my own lab, in terms of energy homeostasis or glucose homeostasis, we need to understand lipid metabolism. And finally, of course, all of you know that on the micro level, the cellular level, lipid metabolism is important, as I will share with you, and even how we regulate organelles within cells and their roles in cellular signaling. So what does that look like in the CAR lab? Well, on the macro level, we're very interested in finding a diagnostic tool for MASLD. Yes, MASLD, no, not MASLD. We don't have that currently. So this is just a snapshot of a slide that's a work in progress that's looking at 888 patients, carefully phenotyped, control patients and patients with MASLD. And we subjected them to lipidomics, and we found a nice signature, we think, that can distinguish those who have MASLD from those who don't have MASLD. So the controls in the blue circle and the cases in the red circle. And by our estimates, we have done really great discrimination, and our AUC is 0.93. Hopefully you'll be seeing that in print later this year. On the meso level, we also have a lot going on in our lab. This is what we look at in physiology, for example. Here is our MET-ALD model, where we feed mice chronic alcohol, this time in their drinking water daily with a weekly gavage, and add to that an obesogenic feeding diet. In this case, high fat, high fructose, high cholesterol diet. And we see what the differences are between the combined condition and the individual conditions alone. So, for example, in our FFC, high fat fructose cholesterol diet plus alcohol, hopefully from the back of the room, you're able to see that this gives the most severe histologic liver phenotype, at least in terms of steatosis, what I'm showing here, and even in terms of early inflammatory markers. We're able also to see from these kinds of studies that glucose tolerance is impaired in the combined condition, perhaps more than any condition, perhaps not statistically significantly different from FFC diet alone, but certainly a trend toward that direction. And this is the curve at the top of this graph, showing that those who are on the FFC alcohol diet, these are mice in this case, have the worst glucose tolerance. And even when we look at plasma leptin levels, which tends to track with adiposity, we are seeing that there's something special about this combined condition compared with any of the other conditions alone. And we're very excited to be able to use this model to ask additional questions. Here in the yellow highlights, I'm showing you that these mice are the most obese of all the conditions, have the highest liver mass, highest adiposity, even ALT levels are significantly elevated. So when we see evidence like this in our lab, and I take it back to my clinic as a hepatologist, I'm already starting to counsel patients that maybe you shouldn't mix that alcohol and that high obesogenic diet together, because the combination may be worse than either one alone. But today we're going to spend most of our time focusing on the micro level, because the fundamental biology is very important and very interesting to me. And we believe that all of these physiologic disruptions ultimately result because of what's happening with lipids on the micro level. For example, our lab has long ago hypothesized that this initial stage of hepatic steatosis, whereby there's an accumulation of lipids within the lipid droplet organelles, is really where all the action is. It's not in the inflammatory pathways, we don't think. It's not in the fibrogenic pathways. Really, everything starts here, where we have an accumulation of lipotoxic lipids that promote oxidative stress, signal the insulin signaling cascade to go awry, And these early changes are what are necessary to promote advanced stages of disease to steatohepatitis, fibrosis, cirrhosis, and beyond. Now, I've said lipid droplet a few times, and I know poster number 6, the presenter whose name I can't remember, knows exactly what I mean. You should go see that poster, by the way. Many of you may not know about the lipid droplet organelle because it's only now gaining some prominence. This organelle is over 3 billion years old. This is actinobacteria, and I'm sure you can see very clearly these organelles are present even in ancient bacteria. And by the way, for the mitochondrial enthusiasts, my organelle is older than even mitochondria, which is merely 1.45 billion years old. And this organelle forms from an outbudding of the outer membrane of the endoplasmic reticulum. This nascent lipid droplet matures into the mature lipid droplet, and every cell has a different composition of neutral lipids. In hepatocytes, they are the triglycerides and cholesterol esters that are the predominant neutral lipid species. These are protected by a thin monolayer phospholipid membrane of multiple proteins and enzymes involved in lipid metabolism, and we have also shown they are involved in glucose metabolism. These are cytoplasmic LDs, not to be confused with the nuclear LDs that, again, poster 6 will tell you all about. These cytoplasmic LDs have numerous fates, including promoting long-term storage of neutral lipids. They can be accessed by lipases to undergo lipolysis. They can be repackaged and secreted as very low density lipoproteins to be circulated outside of hepatocytes. And they can undergo a process called lipophagy, which is the autophagic process specifically for lipid droplets. I'm circling here the perilipin family. Perilipin proteins are the predominant protein families that are the components of this phospholipid membrane. And perilipin 2, PLIN2, in hepatocytes is the predominant protein in this family in hepatocytes and happens to be the protein that my lab's investigations involve. Now, we are very excited not only about the nomenclature change, but even genetics are leading to the lipid droplet organelle as the most important biologic component of SLD. For example, when we look at GWAS studies for MASLD or even ALD, most of the genetic risk is involving the lipid droplet organelle. Here I've adapted a review of MASLD GWAS studies, highlighting all of the genes that have been implicated. And in yellow, I've highlighted the genes that have been implicated in ALD. And what you are seeing is a pattern of genes that are surrounding the lipid droplets or intimately linked with lipid droplet biology. Not much in inflammation, not much in fibrogenesis. So again, steatosis is where the action is. Now, we have added to this literature just last year where we identified 77 total MASLD loci. Some of those had been reported, but we uncovered many new novel loci. And again, most of those genes are in the area of what we categorized metabolism, not so much in the inflammatory space. Now, we have understood for quite a while now that among the lipid droplet proteins in hepatocytes, perilipin 2 is critical. In fact, when we feed mice a Liber-de-Carli diet, this is a special diet that we make every day. It's like a milkshake that has everything in it. We feed them a chronic Liber-de-Carli diet. They normally will develop steatosis, but in the absence of perilipin 2, they are nearly fully protected from steatosis. And when we did glucose tolerance curves, we noticed that normally in the wild type condition, the red graph, mice are glucose intolerant. We talked about that already. But in the absence of PLIN2, whether they're eating a control or alcohol diet, they are very protected. So these were really the first data that linked lipid droplet biology with physiology in our mice. And so we were very interested in understanding how this was happening. We knew a lot already about PLIN2. We know that its major role is like a security guard of lipid droplets. It is protecting the content of those neutral lipids. And it does that by preventing lipolysis, impairing fatty acid oxidation, protecting against lipophagy. We knew a bit already about its regulation at that point that we started by fatty acids. And we have added quite a bit to this literature because now we know that PLIN2 is also regulated by another kind of lipid, bioactive lipids called ceramides. Now, some people ask me, well, you knocked it out, you didn't get steatosis. Why don't we pursue a strategy to knock out PLIN2 in people? And I'm really startled by that question because if you've learned nothing else, this is an ancient organelle that's extremely important. We need PLIN2, but we need the right dose of it. I call it the Goldilocks phenomenon, not too hot, not too cold, just right. So PLIN2 is needed for normal LD homeostasis, but we know that excessive PLIN2 is not good. That's steatosis, and it leads to all the things I showed you. We also know that insufficient PLIN2 or aberrant PLIN2 is not good for us either, likely. Here is a study that my former postdoc, Dr. Scorletti, led, where we took our whole body PLIN2 null mice and transfected them with either AAV wild type PLIN2, which mostly localizes to the liver, or we transfected them with a then noted human polymorphism of PLIN2, SIR251 Pro. We knew that this human polymorphism in experimental models promoted small droplet accumulation, but we didn't know the physiologic effects of this. So here in our mice, we have the wild type transfected condition, fed a high-fat sucrose cholesterol diet. They get steatosis, no surprise there. In our polymorphism mice, they also get steatosis, but they are smaller droplets. In fact, when we quantify triglycerides, they have more triglyceride than even the wild type condition. And so some may say, well, that's great. We've made the droplets smaller. Isn't that what we ultimately want? No, we want normal lipid droplet capacity to store all these lipids so we don't get lipotoxicity. And if we don't have enough capacity, where do those lipids go? Because it's a system. So when we looked at our UK Biobank data of patients who have this human polymorphism of PLIN2, what we found is that they have a preponderance of very large density lipoproteins, and it happens to be the types that are atherogenic. So while we may improve the liver histology, we may be leading these patients to have early heart attacks, perhaps. So that's why it's really important to understand the whole system and the biology of these proteins. So our proposal is that we just figure out a way to tweak PLIN2 to get in that just right zone. And we have found that using ceramide synthetic pathways and what we know about them, we may have some clues coming. Ceramides are sphingolipids. They are comprised of a sphingosine backbone with a varying chain fatty acyl length chains. I'm trying to stay on time here. And I won't go through all of the synthesis, but it's really complicated. I've highlighted two circles here, this ceramide synthase enzyme. It's involved in both the de novo synthetic pathway as well as the lysosomal pathway. And we have now really honed in on this particular enzyme as being key to PLIN2 regulation. In fact, when we looked at mice and even in our cells, the subtype of SIR6 is specifically upregulated in response to alcohol. When we extract our lipid droplets, our cytoplasmic lipid droplets, SIR6 is actually present in the lipid droplet compartment and upregulated in response to alcohol. But in our PLIN2 null condition in the LDs, we actually don't get that same upregulation. So these data suggested to us that it's something specific about the PLIN2 surrounded LDs that SIR6 influences. And in our cells, our VL17A cells, these are HepG2 cells that can metabolize ethanol. Again, normally we would see, this is gene expression in this case, PLIN2 upregulation with the alcohol condition. But when we knock down SIRS6, we don't see that same upregulation. So together, these data demonstrated to us that there's some positive regulation likely happening between SIRS6 and perilipin-2. And in humans, because ultimately we're here, at least my lab is here to help patients, we do find that this is a relevant protein in our patients who have early alcohol-associated steatosis, no inflammation, no fibrosis, and beyond. They have an upregulation of this protein. And in zone 3 hepatocytes, we heard about that already today, where alcohol injury typically first manifests, and we saw the same pattern in our mice. And most recently, my prior postdoc, Dr. Yun, published that in the absence of SIRS6, alcohol-associated steatosis does not occur. Here is a picture, by the way, of our cute mouse feeding a Liber de Carli diet. And with alcohol feeding, these mice are protected from steatosis. Now, again, they do have lipid droplets because we probably wouldn't be able to survive without them. So you can see that from oil red O staining, but it's significantly less. And you can pull up the paper for all of these studies, but I did want to draw your attention to a couple of findings that we found really provocative. When we looked at our RNA-seq data between the SIRS6 null condition and the wild type condition, both fed alcohol, we noticed that two pathways were significantly different: metabolism of lipids, and we were very excited to see lipid particle organization, which we interpreted as lipid droplet organelle biology. And in fact, it actually has a global effect on lipid droplet biology. We saw a number of lipid droplet proteins, including our favorite perilipin-2, that were reduced in the absence of the SIRS6 protein. But we also saw effects on lipophagic pathways. For example, in LC3B2 staining, we saw an increased expression of this protein, suggesting higher lipophagy in the null condition. That was really exciting to us because we had only a couple years before published that ceramides are implicated in lipophagy. For example, when we overexpress this enzyme ASAH that's only in the lysosomal compartment, and when we overexpress that, it reduces ceramides, produces more sphingosine and fatty acids, we found under ultrastructure a number of these lysosomal structures and quantified that here. That led us to think that maybe lipophagy was more pronounced in the condition that lowered ceramides. And indeed, we saw under ultrastructure all of these very beautiful structures and gold stained them with LAMP1 and found that these were indeed lysosomes with lipid droplet organelles inside. Again, higher LC3B2 content. We didn't leave it there. We wanted to know, is this really about lipophagy? When we pharmacologically inhibit lipophagy with lupeptin, we completely reversed this phenotype that had been protective previously. So we do now know that ceramide synthase 6 in part regulates PLIN2 and lipid droplet biology because of its effects on lipophagy. We're also now getting into transcriptional regulation of PLIN2 by SIRS6. And I don't have a lot to present here, but I did just want to give you a sneak peek at where we're going. I've already shown you this top graphic showing the lower expression of PLIN2 in the absence of SIRS6. We also were able to inhibit SIRS6 pharmacologically with FB1 and halt RNA translation. RNA transcription, and found that in the SIRS6 inhibited condition in cells, this does seem to promote PLIN2 RNA degradation, perhaps as one way that SIRS6 transcriptionally regulates PLIN2. When we do our same experiments in our SIRS6 null cells, this time by CRISPR knockout, we are again seeing a similar pattern. In the absence of SIRS6, we're not getting that same upregulation. So we're pretty convinced that SIRS6 is not only reflecting changes in lipophagy. What I didn't show you also, it actually affects the VLDL secretion pathway, but we think it may indeed be a transcription factor. And this is work that my postdoc, Dr. Dempsey, will be taking forward. SIRS6 contains a homeodomain. PLIN2 is predicted to have transcription factors involved in lipid metabolism. And just recently, SIRS2 has been identified as a transcription factor. So he is going to be doing studies to really test this hypothesis. With that, I'll give you the summary slide of all the things we've talked about. Steatosis is where the action is. All of this action is happening long before steatohepatitis, fibrosis, and cirrhosis. Ceramide synthetic pathways are influencing lipid droplet biology, in large part through lipophagy, and we suspect also through transcriptional regulation of perilipin-2. Lipid metabolism informs macro, meso, and micro levels of steatotic liver diseases, and transcriptional regulation is likely. Thank you.
The floor is open in case anyone has questions or if anyone has some on Zoom. If not, I will go ahead and start with one. Super lovely talk. I was kind of curious for your thoughts on breaking down perhaps the SIR6 findings, perhaps others from your lab. What do you think may be shared between your investigations across alcohol-associated liver disease and SLD, or perhaps what do you think may underlie some of the synergistic effects that you showed at the very beginning?
Well, my answer is pretty simple. It is the lipid droplet. ALD individuals get to lipid droplet accumulation differently than those who have MASLD. MASLD is normally through enhanced de novo lipogenesis and higher fatty acids, whereas ALD is predominantly in impairments in beta oxidation, but it all ultimately converges on the lipid droplet organelle, which is why we have focused so intensely on that. I think once it converges there, the pathways are really similar after that point.
Hello.
Hello.
That fantastic talk. We have so much to talk about. I'll focus on one thing. What can you speculate? What's the difference between macrosteatosis and microsteatosis? There's some speculation that micro is worse. How does that play into PLIN2 and SIR6 and all the things that you showed? Thank you.
So what we know is that the lipid droplet protein profile does tend to be different, except we're not talking about the human polymorphism aberrance condition, but in normal individuals, the microsteatotic lipid droplets are enveloped largely by perilipin-3, for example, more so than 2 or even 1 in the severely pathologic condition. So the composition of the droplets is very different. We also see more microsteatosis in conditions of beta oxidative upregulation or even injuries. So for example, women who have fatty liver of pregnancy have microsteatotic conditions. What we typically see, although we say macrosteatosis in ALD and in MASLD, it tends to be more of a mixed phenotype. ALD does lean more toward the microsteatotic because of the beta oxidative impairments, and so they have more, generally have more of the microsteatosis, but most of them are mixed. And I think the macrosteatotic conditions are indeed worse. We know that from liver transplantation, for example. Those livers do not do well without some extraordinary efforts outside of the human body. So perhaps because of some of this biology that I've been talking about, perhaps because those cells even have derangements in the nucleus because they're compressing the nucleus and causing DNA damage. So a lot of things could be going on.
Cool. One more round of applause for Dr. Kark. Our next speaker is Dr. David Lloyd, who is the Vice President of... Let's try again. Our next speaker is Dr. David Lloyd, who is the Vice President of Metabolic Disease at InCitro, where he leads a team that applies machine learning to high-dimensional data sets to generate deeper insights into metabolic diseases for the advancement of high-conviction therapeutic candidates. His postdoctoral studies focused on a forward genetic approach to finding genes associated with metabolic disease.
Okay, thank you very much. First of all, thank you to the hosts and to the organizers. Very much appreciate this opportunity to talk about some of the work that we've been doing at InSitro over the past few years. InSitro is a machine learning company based in South San Francisco. We are very much focused towards using machine learning towards understanding novel pathobiology. Really, we're looking for new targets. Anecdotally, I'd say I've worked in industry for 18 years, and the successes were always fraught with anxiety because every time we moved a target to the clinic, we always had to go and find another target. So this is something that I hope to find use InSitro's kind of platform that they've created so that we can continue to find novel targets in MASLD. So most people are familiar with the declining productivity in R&D. That is that, you know, over the last 50 years and longer, that the number of targets that come out of $1 billion is now down to about 1 drug per $1 billion. This has been termed Eroom's law, which is the opposite of Moore's law. But there does seem to have been a change and an uptick in the last 10 years. And the authors of the last review posit that this could be because of the additional application of genome-wide association studies. And the number of new molecular entities with GWAS is significantly increasing over this period of time. Along with the additional genome-wide association studies, digitized healthcare data, along with genome data, is just being accumulated at an astonishing rate. And I must admit, when I found this figure, I did have to look at what exabyte meant and how many millions and millions and millions of bytes that really was. But needless to say, there's so much data being acquired, there's more genomes being acquired, and this is where we feel we're perfectly placed at InSitro to maximize all this additional data and then apply it to clinical data sets, to cellular data sets, so we can get deeper insights. So this is, as I just mentioned, how InSitro it has been created. Firstly, we acquire clinical data sets, be that through the UK Biobank or through collaborations. We also have, we are doing genome-wide association, we're doing CRISPR genomic studies, automation at scale, live cell multiomics. We have a data platform. I think this plays over here. That's a data platform. And then at the end, once everything is harmonized, we apply machine learning. And through the analysis, we hopefully get greater insights, additional targets, and then we can convert these into therapeutic strategies. So as I mentioned, we apply this across, we use our clinical data, we use cellular disease models at scale, and we combine this with disease learning. And so what I'm going to tell you about for the rest of today is the work that we've been doing in metabolism and specifically in fatty liver disease, here with MRIs with clinical data sets, and here with cellular data sets here looking at quantitative phase contrast images of lipid droplets. So steatotic liver disease. So the opportunity really in metabolic disease was seeing where we could combine these opportunities where we had appropriate data sets for clinical machine learning as well, which we term ClinML. So that was kind of the key and the core focus of where we wanted to start. There's a lot of data in MASLD. There's a lot of biomarkers. There's a lot of imaging data sets. And we are, very familiar from the last conference over the last few days that many opportunities still lie in MASLD. MASLD, I guess it's more of a Z or a Z, depending on where you're from. And in fact, actually, I have to say the take home from that was, it's surprising how few targets there really were that are going into the clinic at the moment. We have thyroid hormone receptor, we have the FGF21. There's a few more in phase 1 early with some SIs, but really there's very few targets on the ground at the moment. Additionally, path to create therapeutics rapidly, SIs, this is quite quick, and feasibility more. Let me see if I can get this right. It's over there. the feasibility to get initial proof of concept. And that isn't necessarily preclinical or clinical proof of concept. That's to prove that the machine learning that we're doing to apply to these clinical data sets is meaningful and has clinical relevance. So this is kind of like our strategy that we use to combine our clinical machine learning with our cellular machine learning. And I mentioned that we acquire data sets so we can do genetic studies from experiments of nature, and we can do genome-wide CRISPR screens in relevant cell types, be they, in this example, I'll tell you about Hep2s, and here we're looking at the lipid droplets. And what we hope to do is by doing enough of this, we can combine the genetics with the genomics and hopefully become more predictive between which cellular models and which genes in which cellular models will be more predictive. And so the validation as we move from these genes and these insights that we find surely should hopefully give us better conviction towards what may work in the clinic. So I'm going to go through two examples now of what we've been doing in machine learning. So the first was based upon a collaboration with Gilead, in which we had access to the clinical trial biopsy data from their ASK1 inhibitor, which is the Stellar 3-4 trial, which is finished now and didn't reach its primary endpoints. Nonetheless, there was a lot of resources from this, and we were able to analyze the H&Es from nearly 7,000 H&E. So we were able to analyze 7,000 H&E stained liver biopsies with no scoring, and applied a convolutional neural net to understand the featureization of those H&Es stained needle biopsies. And I've got really the image here of trying to see the wood from the trees, because when we look at these convolutional neural nets, we're not looking at a specific feature, we're looking at a combination of features. We're not necessarily looking, for what the actual pixel is doing or the pixel next to it, but we're looking for the connections through the pixels. And we try and compress that into an embedding state. And from the embedding state, we can then give it a machine learning fibrosis score. So this became a continuous trait, and this is unsupervised learning. We were able to give a score, an artificial score, to 7,000 biopsies, and then go back and compare those to what did the pathologist say and how did they compare. So the first thing we did is we made sure that we could see correlations between these and that they were falling into the right buckets. And to take it further, we wanted to do some tests for associations. So we looked at fibrosis at baseline, and I think it was 50 or longer weeks later, and see what we could see, the difference between a pathologist score and a fibrosis score. We also did some additional testing on more predictive on RNA-seq and genetics as well. So on the abscissa here, we have the pathologist score for the ISHAT score. And if we just take group 6 here, you can see how variable the ML score was, all the way down to 3. There's some outliers, but there's a really wide range. Now, if you just take this one group out and have a look to see what happened to those patients 48 weeks later, some regressed, some didn't. But it was dependent upon the machine learning fibrosis score was predictive of that based upon week 1. This is 1 × 10⁻⁻10. So, really that quantitative measurement supplied by machine learning was able to tell us, how predictive would the regression be with regards to fibrosis. The other thing we were able to do here was look at the gene expression within samples as well. It's out of sample because it was out of our training set. So we compared a pathologist score to the estimation of gene expression. And so as you can see, for the most part, most of the scores are on the left-hand side here, and this is more of a machine learning embedding prediction score. So the correlations were much greater for the machine learning quantitative score than they were from just the pathologist alone. You imagine These genes here might be more related to fibrosis. And so very clearly, it's hard for the machine learning or the pathologist to be wrong. But we got, in this area here, this is where the machine learning seemed to be more superior. And if we just take this one single dot here, this is HMG-CoA reductase. If you just have a look at the expression versus the expression compared to the pathologist score, there's no correlation. But if you use the machine learning embedding, then the correlation between the gene expression was much greater. And actually 3,000 out of 17,000 genes actually improved in their correlation score as you move towards this machine learning score prediction. And that's interesting how that changed. Okay, so and the next thing we were also able to do was predict genotypes as well. So we've heard a lot about PNPLA3. Everyone's very familiar with it. The GG is a risk variant. This is actually a true pathology histology read, and this is the protective tiles. On the bottom here this is a simulated computer-generated histology score based upon whether it's going to be a GG on the left-hand side or as it moves towards a CC. Again, proving to us that the embeddings were able to predict something. So with the next example, talk about the steatotic imputation. And so we also applied machine learning-based imputation for clinical phenotypes here. And so for some diseases, labeled data is available for a subset of individuals, whereas another measurement may be more abundant. So here, let's just say number of individuals down the left-hand side, they have a lot of input features, but the input labels, such as do they have ICD-10 codes of steatotic liver disease, is not available. So what we're able to do is train using a neural net to predict into the input features and the input labels. Through this training, we can do the imputation, and eventually we can increase the predicted labels. It's so off this pointer as the predicted labels, and get a slightly lower quality in label, but still we've increased the number of people, and this leads to much significantly larger samples and that's well suited for genome-wide association studies. So we applied this to the UK Biobank. Most people are familiar with that. Importantly, that they have a lot of imaging technology there, so we were able to maximize on that. We did use a gold standard PDFF as some of our training labels, and we were able to predict and impute the alternative data sets. So here we're using the ML imputation applied to MRI-derived liver fat. So as I mentioned in the training example, here we have a lot of individuals. There's actually 262,000 with genetics and only a few with liver fat imputed. So, and then there's a lot more individuals that have either metabolites or biomarkers. And through the ML imputation, we're able to increase this data set. So that gives us greater conviction from multiple modalities. Sample size increases for statistical genetics, and we converted this to a quantitative trait as well, which really improves the genetic discovery. So we weren't doing genome-wide association studies on 25,000. We ended up doing genome-wide association studies on 262,000. In the interest of time, I'm going to skip this, but I will show you this is what the association was like. So this was true liver fat on the bottom, and this is predicted based upon 37 biomarkers. And so the R squared is quite low, but still there was a bit of a solar system kind of cloud to this. And so we did feel it was moderate, but we still wanted to pursue it. To improve our conviction here, we wanted to do some ablation studies. And so that's kind of remove a few biomarkers at a time or clusters of biomarker to improve your conviction towards it's not what you predicted in liver fat. It's not just driven by a particular trait or one particular biomarker. And you can see through all 37 biomarkers, sometimes they cluster together like these adiposity related ones. Obviously, if a waist circumference is probably going to be quite close to a BMI. And so the predictions were all relatively good between the full model and the ablated model. The one that we saw the most adjustment for was the adiposity markers. And so we're going to do genome-wide association studies on that now. But these are the, actually, I just quickly mention about what do we know so far about the genetic landscape of MASLD from common variants. So, you know, since the PMPLA3 association's been out for, you know, nearly 15 years now, there has been a trickle of more loci that have started to come in. The most recent, which is probably the largest meta-analysis, was done by Decode. And you can see here, this is a typical Manhattan plot, and you're very familiar with this cluster towards 19 and 22 with the PMPLA3, the APO3s, and the TM6SF2s, PMPLA3. So what we actually found through our work is that if you just go from MRI PDFF and now move to biomarker predicted liver fat, this is the change in the landscape. The Manhattan plot drastically changes. And you see some of these genes like involved in de novo lipogenesis are increasing. Some of the genes like PMPLA3 and those regulating the lipid droplet are decreasing. And actually, I thought a real kind of comforting effect from this was the fact that we believe that we've got the first genetic association to liver fat at the FGF21 locus. And as you know, that's being pursued quite actively in phase 3 at the moment and seems to be performing quite well. I think the one thing that we wanted to do, because we were using a proxy phenotype, it was machine learned, and it was just on liver fat, it wasn't really clinical disease. We wanted to understand, you know, how did this compare to, how did this correlate with clinical NAFLD? So the geneticist, Harry, took the in situ sum stats and the z-scores, compared those to the ones from Decode, which is the study that I presented to you, and compared them and observed that there was really strong correlation between the effects of all the variants, including those that were not genome-wide as significant. So anything that was less than 10 times 10 to the minus 8. Importantly, MRI PDFF is very closely genetically correlated to the Decode data, but the data that we got from biomarkers is, if not as good, if not better. So what we really think we found here is, because of the number of loci, we increased it, you know, almost increased it by 100. We think we've got a completely different view of what the genetics is telling us about fatty liver disease. So based upon prior genetics, not biology, but genetics, there's probably 4 genes involved in the lipid droplet. There was a few genes involved in de novo lipogenesis and perhaps a few in TG export. But now with these new findings, we really think as though we've got the genetic data that pushes us in the direction to say de novo lipogenesis really is the key behind MASLD. MASLD, got it right. Okay, so, and then we do actually have a novel gene out of this. We're pursuing it at the moment. It's getting, some therapeutics are getting dosed in mice in a few days. But the one that I want to point out is that we've got, if we just use MRI alone, it was not significant. Metabolites, it increases its significance. And we use biomarkers, the p-value is kind of off the charts, really. And in doing some meta-analyses with other data, we're starting to reach genome-wide association significance, even in clinically diagnosed MASLD as well. So with a minute left, I just want to tell you about a complementary approach that we're taking in terms of genomic approaches, and that is we're doing CRISPR-based screens, but we're doing pooled screens. So this one is a druggable genome. We're actually looking at ablating every gene in the full genome, the whole genome, and it's pooled. So every gene, every neighboring hepatocyte will have a different knockout in it. And it means that we can do a genome-wide screen in a couple of 6-well plates. We're able to deconvolute that by sequencing the actual individual cells. And you can see this is sequencing the cells, residue by nucleotide by nucleotide, and then corresponding the target with, or the gene that's been knocked out with the phenotype. And this is what we've been doing at the moment, finding the gene and then also correlating that to the phenotype as well. I think just to push forward, this is the results of our first screen that was just recently completed. We've got differences. So we look at different phenotypic readouts, BODIPI cell intensity, number of lipid droplets. We'll also use machine learning to compress, orthogonal partial least squares on multiple aspects of cell morphology. And we're able to identify, the common players in those that revert the phenotype when loaded up with BODIPI, or those that exacerbate the phenotype. And just to bring it full circle, what we want to do is that now, the first screen I showed you was loading hepatocytes up with fatty acids. We will want to convert this into a de novo lipogenic screen as well. So in conclusion, I told you about this kind of spiral that we're applying and how we're integrating the human genetics with the cellular genetics. And so I told you about how we're using the biobank and we're imputing phenotypes and creating machine learned phenotypes. We're doing genome-wide association studies. We've got additional insights now into the genetics behind steatotic liver disease, and we're converting that into genome-wide screens using CRISPR technology. So with that, just like to thank the team that has been doing all of this work, and thank you very much for your time.
Thanks, David. That was really nice work. I think AMRA is doing body composition imaging of about 25,000 UK biobank subjects. So it might be interesting, that includes visceral adiposity imaging and sub-Q. Be interesting to, when that data becomes available, maybe layer it into some of your circulating adipocyte markers.
No, absolutely. Yeah. And I think the key is to go bigger as well. So.
Can I? Okay, thank you. Wonderful presentation. I was wondering now, your system is trained to see the clinical, preclinical data and all of that. Would you be able to predict different approaches within the same modality? For example, FGF21 versus thyroid receptor beta, whatever, So would you be able to predict, knowing what you have trained your system, to say which one has highest probability to achieve clinical success at a various spectrum of the disease? That's one. And can you also distinguish from other modalities, cell therapy, things like that?
So that is the key question. Can the in vitro or maybe preclinical be predictive of clinical success, really? And can you take, so what insights can we get from the genetics first? Because that's already available. And I think in steatotic liver disease, there's so many clinical molecules available at the moment that we can kind of do the back translation and see, can we create predictive models? So I absolutely, completely
that's
agree.
where the real value is, right?
Yes.
What you put in, quality, high quality, you put in high quality machine learning, you'll get in. But the predictability of the different modalities and within the modalities, different agents would have a high value, I think.
No, absolutely. I think trying to segregate the different aspects of the biology as well, and then trying to bucket those pharmacotherapies would be a key way to diversify a portfolio, for example.
But at the moment, you don't have that capability, but you're thinking to develop.
No, we are doing it.
Okay, thanks.
Thank you.
And it is my great pleasure to introduce Dr. Irwin Arias as our final speaker for the day. He is a triple emeritus at Albert Einstein College of Medicine, Tufts University School of Medicine, and senior scientist at the NIH, where he continues to teach. Some of his most notable accomplishments include mechanisms of inheritable jaundice, bile transport, and hepatocyte polarization. These research outcomes established new paradigms for biliary secretion and inheritable liver diseases. As evidenced by the 32nd Arias Symposium that we're all here today, he has dedicated his career to bridging the gap between scientists in the lab and medical doctors in the clinic.
Are you going to advance the slide? Well, I want to thank you all for being here. I want to thank the American Liver Foundation, Sangeeta, Wolfram, Heather. I always find these as one of the most exciting meetings I ever attend to, because we really get glimpses into the future, not so much the past. And the future comes along very, very quickly, more quickly now these days than it did not so very long ago. So I want to talk to you a little bit different from what has been presented before. I want to talk to you about something that is biologically extremely important, essential for life, and totally neglected in the liver field. And so this is more in the nature of whetting your appetite and perhaps stimulating some of your thoughts and research into a huge problem, which is the issue of cell polarity. Now, polarity is very ancient, probably going back to the Mesozoic era, when a couple of cells got together and eventually formed something like a clump or an organ. And if you didn't have polarity, just think of it, you would never be able to separate the inside from the outside of things being pumped out and other things being absorbed. So you'd never have organs, you'd never have organisms. As a matter of fact, you'd never have life. So polarity is present in probably every cell in our body, with the possible exception of the erythrocyte. And all of these cells have a common blueprint, which is established during their embryologic development, where there are three major complexes of largely proteins that determine what is like an apical domain from a lateral, from a basal domain. That blueprint is present in every cell, except maybe the red cell. But every cell type superimposes upon that a kind of a recipe to create its own nature of polarity. And so you wind up with different forms of polarity. So, for example, shown in this slide, you see a neuron. That's highly polarized, of course. You look at a T cell. You can look at a parasite. And for today's purpose, you can look at a epithelial cell. So these are different recipes. But what are the recipes? Move to the next slide, please. Now, hepatocellular polarity has four features I would like to briefly emphasize. One is it is totally neglected. The second is it's unique, as I'll show you. It's highly complicated, and it's essential for function. So let's look at those four things. Neglected. Believe it or not, no, that's not the next slide. You jumped one. But let's have the next slide. Please, the next slide. Yeah, okay. There's only one textbook, and I'm not advertising this, in which the word polarity appears, let alone any discussion. There is no textbook of pathology, medicine, gastroenterology, or hepatology, -1, in which the word polarity appears anywhere in the text. It's quite amazing. Why is that? Now let's go to that slide. I think I know the reason why. This is a hematoxylin and eosin stained section of a human liver. Now, the H&E stain was developed almost 100 years ago, and it's the workhorse of pathology. And pathology has been the major discipline that really sort of brought hepatology into being. Well, if this is the major cell stain that's used for diagnostic purposes, something's missing. You can't find the apical domain of hepatocytes in an H&E stain. In fact, for many years, they never even knew what it looked like until electron and transmission microscopy came along and demonstrated a capillary-like structure that runs between cords of hepatocytes, forming a network. And that capillite structure is the apical domain of the hepatocyte, across which every substituent of bile is secreted. If you don't see it, you don't comment on it, and at least that's my theory as to why in no pathology report I or any of my hepatology colleagues have ever seen that the pathologist says, aha, the hepatocytes have lost their polarity. It doesn't exist. So it's neglected. Next, please. Next slide, please. So it's unique. As shown here, the polarity in ductal tissue, the bile ducts, pancreatic ducts, also in the intestine, the respiratory tree, the nephron. It's lined up so there's a central luminal cavity. The cells have their polarized domain that forms the central cavity. But the hepatocyte is unique because it's the only situation in the body where two cells come together and almost by magic align between 8 and 10% of their plasma membranes together to form a tube, a capillary-sized structure, which is sealed by tight junctions and forms a network. Stained here, and you see it as the bile-canalicular network. Only so in the body, truly unique. Next slide, please. Now, from a transporter's point of view, we can look at this diagram. And the canaliculus is the central portion there, and it's the home to a large number of proteins, most of which are ectoenzymes, but also a home to a family of ATP binding cassette transporters, which hydrolyze ATP and conduct active transport for all of the substituents of bile, organic anions, a separate one for cations, a separate one for phospholipid, and as shown in red, a separate one, ABCB11, for bile acids. Now, bile acids are nature's detergents. They're created in the liver from cholesterol by the action of 7-alpha hydroxylase, and they're intensely regulated by various transcriptional events. Well, bile acids are secreted, and after a meal, I'm sure you know that they are reabsorbed into the circulation and come back to the liver in a big wave. So we ask the question, how does this transporter and the other ones by inference selectively go to the apical membrane? And nobody knew, really. Next slide. So this slide is 25 years of work at Einstein, at Tufts, at the NIH, with a large number of colleagues, students, fellows, and people in a variety of disciplines. In the last 15 years, outstanding cell biologists, particularly Jennifer Lippincott-Schwartz and her group, of which I was privileged to be a perpetual postdoctoral fellow for 15 years while we studied these things. So the techniques that were used were histochemistry, immunocytochemistry, inhibition with various drugs or activators. And over the past decade and a half, most advanced imaging techniques, including those for single molecules. So this involved the use of chimeric green, yellow, and red fluorescent ABCB11. Then we progressed into super resolution microscopy, and more recently actually have been using in collaboration with a very brilliant woman at NIH, Natalie Porras-Leon. We've been using supravital microscopy, observing this pathway actually in the liver of living mice that are normal and also transgenic. So I'm going to take you on a tour of what we think a molecule of ABCB11 undergoes. And this roadmap may seem to be rather complicated, but I'm convinced in my soul that this is really still scratching the surface, because new members and new pathways begin to feed into this magic event of polarization. So ABCC11 is transcribed in the nucleus. There are several diseases, mainly in children, where there are mutations of ABCB11, misfoldings of ABC11, and a modest amount of information exists regarding the transcriptional regulation of this protein itself. From the nucleus, it's transferred, of course, into the endoplasmic reticulum, where the protein is synthesized, undergoes proper folding, and acquires at least 2 sites which are N-glycosylated. And that's required for it to then bind to a RAB11 GTPase and a motor molecule, myosin 5B. And it's this complex that moves along the microtubular network throughout the entire cell. The entire cell. It moves to the lateral, the basal, and the apical. But it does not attach to any of the plasma membrane except the apical domain, because that's where a docking protein, syntaxin 3, resides, and that's the binding site. So the transporter is attached to syntaxin 3, which brings it in the apical membrane, in which it diffuses throughout its entire thing, but the diffusion is limited by tight junctions that prevent its diffusion into the lateral plasma membrane domain. Now, a remarkable thing is that most of the ABCB11 is not in the apical membrane. It exists in a large RAB11A pool within the cell, from which it is continuously cycling on and off the apical membrane. And after you ate a meal, when a big load of bile acid comes back up in the enterohepatic circulation, the pool shifts to the apical domain, and we understand something about the mechanism of that, to accommodate this load. And after the bile acid load has been secreted, it then goes back to its endogenous state. Now, in addition, there are pathways that are involved both in the recycling endosomic process and also in an endosomal type process that leads to the degradation of the protein. All proteins eventually are made and degraded. And just shown in red here, some of the components of this, which were demonstrated by Daniel Ortiz, who's sitting over here. HAX1, which is linked to cortactin, and the EPS15 system involved in recycling and also connected to clathrin-mediated endocytosis, which presumably is a major route leading to the protein degradation. Now, this entire process is dependent also on the presence of the PAR complex of proteins, which in turn regulate tight junctional formation. We'll get to that. And the entire process is energy dependent. So it's dependent upon the energy generated by oxidative phosphorylation, but in the presence of stress of various kinds, there is activation of two protein kinases, LKB1 and AMPK, which selectively keep this polarity system alive, even though the ATP levels in the cell may be quite low and have not been restored to normal. So let's look at a little of this. Next slide, please. So we ask a question, are these many components, which one's critical? Can the cell carry out polarization if you block the tight junction? Or if you inhibit microtubular trafficking? Or if you affect this recycling endosome system? Or for that matter, if you alter energy? And through a period of many years and many inhibitors, silencing RNAs, various drugs, and as shown in magenta, there are a series of conditional knockout mice. The most recent one, performed by a group in Pittsburgh, involved the beta-gamma-catenin system. All of these lead to altered polarization. The polarization is not all phenotypically identical, but in the case of the liver, if you alter ABC trafficking to the apical membrane, and the cell overcomes its limited ability to get rid of bile acids across other membranes, or even to help modulate its synthesis, the detergent accumulates within the hepatocyte and produces cellular damage, particularly to membranes, and especially to the mitochondria. And so it leads to mitochondrial fission. Mitochondria appear very small, and their capacity to synthesize ATP is grossly restricted. So all of these are part of a majestic tapestry. Next slide, please. And here's the tapestry. So without proper trafficking of the transporters and associated lipids, which we didn't talk about, without the cytoskeletal microtubular system and its connectin to actin at the apical domain, and without the tight junctions in particular, and especially with restrictions of energy metabolism, the result is depolarization. But they are all required collectively in order to produce the beautiful biological picture. And this is only an incomplete view. I'm sure there are many other aspects that have yet to be explored. Next slide, please. Well, let me show you an example of the more recent technology that we've been using with Natalie Porras-Leon, and that's supravital microscopy in living animals. So what we have here, let's see, it's on your left, is a view into the liver of a living mouse. And what you're going to see, I hope, but I can't turn it on from here. There we go. So the black area is where the hepatocytes are. Nothing's going on there. And remember, I told you between two rows of hepatocytes is the thin bile canaliculae. So you don't see anything there. What you do see is red and some gray. The red is 6 kilodalton rhodamine dextran injected into the animal, and that's the circulation. That's going through the hepatic sinusoids. The gray, don't pay much attention to. We were experimenting with some small, sort of like microtubes to see if we could use that instead of rhodamine. The point is the area in black. Now, to your right is what you see in a conditional knockout of LKB1. So this is a protein kinase activated by another protein called STRAD and requiring a stabilizing protein. And when it's autophosphorylated, it phosphorylates a very specific site on AMPK. Now, AMPK is the metabolic sensor within the cell, and it responds to falls in ATP when an increased rise in AMP by comparison. And the net effect, it shuts down every system in the liver that requires energy, with the exception of one, and that's polarity. It's almost as if ancestrally, maybe going back to the Mesozoic era, polarity being such an essential part of life, and this is my hypothesis, it essentially is protected, even in the face of marked decline in oxidative phosphorylation and other energy metabolism. Okay, back to the slide. Now you see in the black area, there's red. That red is 6 kilodalton rhodamine, which has gained access to the biocanalicus through the tight junction, which in the LKB1 animal, knockout animal, the tight junction has lost its permeability restriction. And in large part, it's due to the fact that there are 2 proteins, part of the tight junction, which require specific phosphorylation, and that's carried out by AMPK. There also is a specific phosphorylation site, CLIP170, at the end of the dynamic microtubules, and LKB1 controls that. And if that site's not phosphorylated, the microtubular system stops. So the LKB1 AMPK axis is an important regulator. So here's a beautiful example in vivo, not in cell culture, which is what most of our work was, as I showed you previously, that LKB1 regulates this paracellular pathway. There are now things can get from the biliary space back to the blood, and from the blood to the biliary space. That's never been seen before. It's been postulated and tested indirectly, but here's direct vision. Next slide, please. So here is quantification of it. The red shows the amount that appears, and in the normal, in the black, nothing's there. Next slide, please. Now, when I talk about polarization, I have to explain to you what we mean and what we see experimentally. So the apical domain contains a bunch of ectoenzymes and these ABC transporters, a lot of them. It contains syntaxin 3 and the tight and adherent junction proteins, which are regulated largely by PAR1. Now, when depolarization occurs, this is complete depolarization, all of these components appear someplace else in the cell. Many of them appear in the lateral or basal plasma membrane. And whether ABC-B11, for example, if it's in the lateral membrane or the basal membrane, whether it's doing anything, we don't have the faintest idea. But in addition, all of these appear within cytoplasmic clusters, some of which are undoubtedly degradative, but most of them have not been identified. Now, what of great importance is the result of this process, as it evolves fully to depolarization, is impaired cellular function and eventual cell death. So, polarization is pretty damn important. Next slide. Part 4. Polarization is important in human disease. So here are candidate polarity diseases. Could it be an inheritable liver developmental diseases, of which there are many? What about the hepatic mitochondrial defects of inherited kind or those associated with drugs? What about the metabolic stress that's produced by things like enterotoxin, which we showed inhibits AMPK mitochondrial dynamics, and others showed it too. Another candidate are disorders that are characterized by substantial bile acid retention, such as with drugs or with viruses. So here are the candidates. Next slide. Now, there are now 11 Mendelian inherited hepatocyte polarity disorders. And they're all shown here, and I'm just going to show you one example, which is perhaps the most dramatic. Next, please. So The example I'll show you is called microvillus inclusion disease, also called Davidson's disease. This is a recessively inherited disorder where children are born and they're incapable of absorbing anything pretty much from their intestine. And they also have marked liver and sometimes kidney abnormalities. In the liver, it's called bile secretory failure or cholestasis. Now, when pathologists looked at the small intestine, they see that the brush border, instead of being up on top, next slide, please, is actually like clustered in the cytoplasm. So they called it an inclusion disease. The point of it is it's the brush border in the intestine and the comparable microvilli on the surface of the canalicular or apical domain, which are the place where the action is taking place to secrete and to absorb. In some individuals, most of them, the GI tract manifestation is the most devastating. In others, it's rather negligible and the liver is more demonstrating. And in very rare ones, the kidney is. And why these variations are completely unknown. These are all due to mutations in the myosin 5B gene, which you remember is the motor molecule that's driving ABCB11 from the trans-Golgi to its site in the apical domain. And it's probably driving many things of that nature to the apical domain. Next slide, please. Now, here, to my way of thinking, is the $64 million question. Do acquired liver diseases result in loss of polarity? Now, you would think, we should know that. After all you have to do is take some liver biopsies and stain them for some of the ectoenzymes for which there are antibodies in abundance, or for one of the transporters for which there are also antibodies. But nobody has ever done it. And it beats me to understand why hepatologists haven't jumped on this to address this question, because I think it's likely, given all the abnormalities that are associated with liver metabolism, cholestasis, and everything else, polarity, I think, has got to be an important and maybe even the focal point related to the pathobiology of those disorders. And it shouldn't be so complicated to actually study. Now, maybe there's a student here or a postdoc or someone who gets excited by this and says, hey, look, it's worth taking a look seriously. I'm sure it's a little more complicated than I've made it out to be. Now, next slide. So I think we're on the verge of a great discovery, that E equals MC squared. And it's just a question of applying modern advanced cell biologic techniques and even simple enzyme histochemistry to begin to address this question. And I am pleased, last slide, to announce that we are going to have a breakthrough. The Howard Hughes Medical Institute recently agreed to host this coming fall in 2024, the first international workshop on cellular polarity. It will be international, it will be multidisciplinary, and it will be very exciting. And we hope when you see the notice that you will submit an abstract, because we will encourage students, fellows, and everybody to do that, and to come and to enjoy. It will probably be about 3, maybe even 4 days of talks, but mainly of conversations. So we hope we'll all see you at Genelia Farms in 2024. So thank you all for being here. Marcos?
I have a kind of a teaser chicken and egg question. So your bile acid transporter life cycle is great, but it depends on syntaxin already being in the apical membrane for it to dock. So syntaxin comes before ABC11. How does syntaxin know
to go to the apical membrane?
I'm sorry, say it again. Syntax.
How syntaxin is in the apical membrane. It needs to be there as a docking spot for the APSC complex. So clearly, syntaxin going to the apical membrane precedes ABC11 getting to the apical. So how does syntaxin know to go to the apical membrane?
Interestingly enough, to my knowledge, no one has ever done the kind of dynamic cell biologic imaging that I'm talking about with respect to syntaxin. It was identified as an association protein, its localization, its binding, and its requirement. There is a syntaxin knockout in cells and in a mouse, and the whole mechanism, doesn't act normally. Things go all over the place with respect to B11. Now, what is syntaxin as a molecule? I just don't remember or never knew.
Wayne, that was a beautiful talk. You're such an amazing teacher. My question is about hepatitis C virus and how occludin-1 and claudin-1 are thought to be entry receptors for the virus. And I was curious if you had any thoughts on how things get in via the apical domain.
I couldn't quite understand what Sangeeta was saying.
She was asking about occludin-1 and claudin-1 in the context of hepatitis C virus and how those are the entry receptors.
Oh, that's this field is loaded with challenging questions because we know so little. These, of course, are sort of binding uptake sites for the virus. To my knowledge, there's no information whatsoever. This is an example of bridge building. There is no linkage that I'm aware of between, for example, this kind of cell biology and, say, the virology of entry. The site of what they're attached to is fine and identified and so forth. But the interaction, the dynamic interaction between these, I think, has probably not been very extensively explored. So I can't answer that, Sangeeta. I don't know.
One more round. Oh, sorry. Was there one last question? All right. In that case, one more well-deserved round of applause. Thank you for a very lovely. And now I will hand it off to Heather.
I need something. Can I? Yep. Okay, great. Hi, everybody. I'm the Heather that sends out all the emails. I didn't put myself on the program because otherwise I would talk too long. So I am just going to appear and thank absolutely everybody for being here, especially the session chairs. It's a hard job to be here, especially on a hybrid format. We're all just figuring this out as we go along, and they did a great job. And so we're really pleased to have them here. I am mostly here to present the poster prizes, but actually I also wanted to thank everybody on the Zoom. I have no idea if they can see me at this point, but oh, maybe they can. This was really a tough experiment, but I'm glad we did it because I think it gave so many more people an opportunity to hear just all the fabulousness that we got to experience in person. And I would invite everybody on Zoom, if you're able to come in person next year, it gets even better if you're here in the room. But we're really glad that we did it. And so now I will very quickly, because we're behind time, and I really want a glass of wine. I want to celebrate the poster prizes. So I was really glad this poster, it was an experiment we did in 2019. I wanted to do it again. I think it's really important to give the opportunity for trainees to talk about their work, and with just so many people from so different backgrounds. And for those of you who weren't involved in the judging, we actually had 16 people helping out with the judging. And we had pairs of people, again, with the bridging concepts. There were academics and clinicians and people working in industry, and they all got to judge together. And it's a great experience in different sides. So all that being said, the scores were very, very, very close. It's hard to actually name winners, but they're numbers, so we can do it that way. So first, honorable mention with no prize, unfortunately, goes to Amy Stoddard from the Chen Lab. But I had to, her score was so close that I had to call her out too. Hopefully she's still here. And then the third prize goes to Prashant Nagesh. Not sure if he's still here. If not, I'll hold on to his prize. A lot of people had to go back to lab, I think. And then Anna Smith from the Gouon-Evans lab. Is she here? Yeah, come down. And oh, I don't have them on my phone. There is Anna, do you remember your title?
Yeah, it was transient expression of HGF and EGF using mRNA and LNPs promotes engraftment of primary human hepatocytes in murine liver disease.
That was long. Good job. And then our top prize goes to someone whose name I never, Constantine, I don't know how to pronounce your last name. I just know you as Constantine. Constantine from the Shalik lab. Tell me how to pronounce it. Chuanis, Constantine Chuanis, thank you. Oh yeah, tell me your title.
Title is something along the lines of chronic stress adaptations drive trade-offs between development-associated phenotypes and tissue function in non-transformed liver.
The notes from the judges, the only criticism of Constantine's poster was that he had too much data. It's a nice problem to have. And then lastly, for the fan favorites, there was a tie. So we can welcome both Patrice Delaney and Janvi Huria. And then maybe I'll hand these way behind.
Okay. All that is left for us is to give out a lot of thank yous. The first one is to all of you in the room and everyone on Zoom for being here with us and forming the community that we all so cherish. To Wen for essentially inspiring all of us over all these years and bringing us together. And in just an incredible way, at the end of our day, pulling it all together and teaching us. What an incredible gift. Thank you. Heather, I know personally that this would have not happened just with Sangeeta and myself. It's all you, and to your spirit and your tenacity and you prodding and pushing us, making this a reality. It's just incredible, and it's so great to be able to work with you. And I hand over.
Yeah. You're here. You're here. Thank you for everything. And then just to wrap it up, Matt Perkins, who's I think here behind the door. Yes. Dealing with our experimental hybrid format all day. Thank you. Sarah Bowen, and the whole ALF team for everything. So we're so delighted with this continued collaboration, which started some 40 years ago at Wynn's urging, and the 32nd symposium. I declare happily over and a success, I think. So we're going to retire to the wine, and thank you all for spending the day with us.